Hello, good morning to everybody. Welcome to our online conference, Shaping the Future of Micro and Nanoplastic Research, Connecting Science, Policy, and Industry for Human Health, that is organized by the PlasticHeal project, and that will run today and tomorrow. It is now evident that micro and nanoplastic pollution represents a health to human health, represents a threat, to human health and the environment, and that there are many actors involved with this statement in two directions, meaning that we are all part of the problem and also part of the solution. And it's more and more evident that it's very important that we sit together to co-create different strategies to reach the ambitious goal of having a better environment and healthier lives. And this is the exercise that we are trying to do today, sit together with this objective. Out of the many different strategies that one can do to reach this goal, one is to have better preventive policies and regulatory initiatives. And PlasticHeal is aligned with this potential way of finding a solution to the problem of plastic contamination. So the main aim of PlasticHeal is to try to make all the information that has been generated in the project and all the technologies that have been generated within the project useful for human risk assessment and regulatory purpose. Just to give you a quick overview of the project, so you have a little bit of context. PlasticHeal considers that there are main sources of environmental exposure to micronanoplastics, which is food and air. So humans are exposed through inhalation and ingestion, and these microplastics go to the primary barriers, the respiratory and the gastrointestinal tract, then translocate to the bloodstream, and then distributes to the secondary organs where it can persist through time. So what we do in PlasticHeal is to develop different approaches to cover each one of these steps in the chain of the micro nanoplastic journey. The first is through biomonitoring studies, then having models of different complexity in vitro and in vivo, although we rely mostly on in vitro models of the primary barriers, blood system, and secondary organ. And in all of these models, the idea is to measure a bunch of different effects, from genotoxicity to respiratory and gastrointestinal toxicity, immunotoxicity, carcinogenicity, etc., that will be measured across all the different technologies that we have developed. So we will generate a lot of information, and the project is arriving to its end. It will finish at the end of March next year. So most of these models that I was describing and most of the data that we meant to collect are already collected. We are now ready for doing a human risk evaluation with all this information. And the idea is to share this with you so we can incorporate your input and maximize the project impact. So from now to the end of the project, we will do this exercise twice. One now with this conference, we will incorporate all the input collected to our project, and then we will share it again in the context of the CASP cluster where we belong, and where we will work together with other projects with similar objectives. And then we will incorporate again the input collected within the cluster with the rest of our collected projects. So today we will have the opportunity to share and discuss and collect input about all the information that we generated and the framework, the conceptualization that we have prepared to collect and evaluate this information. And tomorrow we will have the chance to discuss about the technologies. And the idea is that we keep in mind that the aim of the whole discussion is to maximize the value of the project's data for risk assessment, human risk assessment and regulatory schemes. And with this introduction, I will just pass the floor to Virginia so she can give an overview of the agenda and describe how the session will be developed.
Good morning, everybody, and thank you for joining us today, and thank you, Alba, for setting the stage for today's session. My name is Virginia Mata, and I lead the communication efforts for the PlasticHeal project. And as Alba has explained, PlasticHeal is an EU project, EU-funded initiative focused on understanding the impact of micro nanoplastics. Some of you are familiar with this project, some of you don't. For those of you new to the project, welcome. So over the next 2 days, we will explore how the cutting-edge research, innovative methodologies, and collaborative approaches are shaping the future of micro nanoplastics, risk assessment, and regulation. So let me please give you a picture of who are joining us today. Today's audience brings together an incredible mix of professionals, academics, regulators, and industry stakeholders. Each one of you are playing a critical role in tackling the challenges posed by micro nanoplastics. We hope that this event fosters meaningful discussions and insights and collaborations, of course. We're honored to kick off the day with a keynote presentation by Steffen Foss Hansen. He's a professor at the Technical University of Denmark and a key contributor to the PlasticHeal project. He will present the framework for risk assessment developed by the PlasticHeal team. But before we begin, let me give you a quick note on today's format. The session is being recorded, and the video will be shared on YouTube and social media afterwards. Following each presentation, we will have 5 minutes for technical questions. Only, please, technical questions, because by the end of the day, we will have enough room for people to debate. It will be an open debate for all of you to participate. And please feel free to submit your questions via the chat for these 5 minutes after each presentation. We have also set up a shared whiteboard that I will activate after each presentation, where you can leave comments, share ideas, or raise broader questions throughout the session. We encourage you to use this space actively, and it's a great way to foster dialogue and collaboration. And lastly, while you may not see it live, a graphic recorder is capturing the key points from today's session. And these visual summaries, along with other materials and minutes and so on, will be shared with all the attendees in the coming days. So thank you for being here, and let's dive into today's program. Please join me in welcoming our first speaker, which is Steffen Foss. Welcome, Steffen.
Thank you very much. I'll just share my screen. And then we should be ready to go. So the topic of my talk today is the risk evaluation and categorization framework that we have developed as part of the PlasticHeal project. And of course, even though it's my name on the first slide here, it's part of a collaborative work that we have done in the project in the last 3 years. And I look very much forward to introducing it to you, and also to discuss it with you further during the day. So we've seen a dramatic increase in the number of scientific publications on nano and microplastics in recent years. At the moment, there is pretty much an increase of about 6,000 to 7,000 papers per year. And that has to be put in the perspective of the fact that 15 years ago, there was only about 305 papers on the topic. So the increase has really been very, very dramatic. When it comes to the topic of today, which is public health and toxicology, there's almost 20,000 papers on or relevant to the topic of nano and microplastics. So of course that raises a fundamental question is how do we actually create an overview and understand all that information that's being generated. Like, we know more and more and more, but our understanding is not necessarily able to follow. And that's a significant challenge that we have faced in the last 3 years of the PlasticHeal project. When we look across the scientific studies that have been published, we can see that all kinds of different adverse effects on human health or have been reported, especially in laboratory studies. Alba alluded to it a little bit previously, but PlasticHeal is part of a cluster of projects that study the human health risks of nano and microplastics. And there's 5 of the projects. Across the different projects, we have published more than 60 scientific papers and also a bunch of deliverables. If we look at both the general literature and as well as the scientific studies that have been published in the CUSP projects and in PlasticHeal, we can see that effects are reported related or relevant to respiratory toxicity, gastrointestinal toxicity, secondary organs, reproductive toxicity, effects on immune systems and DNA. And we also see long-term effects or accumulation of nano and microplastics. So the key challenge that we faced was how do we actually include and communicate all the information and all the knowledge that is being generated, not only in our project, but also in general, in a way that it's meaningful for risk assessors and regulators. And the idea that we came up with is what we call the plastic risk categorization framework of PlasticHeal. And basically, it's a radar plot that looks like this. And I'll spend the rest of my my talk here on introducing you to all the different elements of this framework and this idea. And then I look very much forward to receiving your input on this and sparring on different ideas that you might have that we have time to pursue in the coming months before the project ends in the end of March next year. So the first element to explain or to dive in a little bit into is in the top of the radar plot, just below the logo of the framework. And here we're defining the scope of the evaluation. You can see here that we've inserted a polymer 1, and that could be any kind of a polymer that you're interested in. It could be polystyrene, PET, whatever you can imagine. And that's kind of like indicated by the red line. That is the polymer that, or whatever it might be, that we're interested in evaluating. Of course, the scope of the evaluation can vary a lot. So here on this slide, you can see 4 different examples of varying scopes of the plastic risk cat evaluation framework. So in the top left corner, you have the one that we also had on the previous slide, where the evaluation or the subject to evaluation is just one individual polymer. But you could also imagine that we wanted to create evaluation that illustrated the results of several different polymers, and that you would just also explain just beneath the logo with having different polymers represented by different line colors. You could also imagine, as you see in example C, that you have one given polymer that you know has different additives in it, and you want to illustrate the combined knowledge of the health effects of both the polymer as well as the additives, and that you could also illustrate in the plot. And then finally, you could also imagine that you have a polymer and you have a lot of knowledge about how this polymer interacts with different kinds of environmental pollutants, and you could then also illustrate that. So it's just important that when you want to use the evaluation framework, that it's very clear from the outset what is the actual subject of the evaluation. A second key element of the the evaluation framework that I'm presenting here today is the category labels that we have decided to put on the radar plot. So if you can see that we have like carcinogenicity, we have reproductive toxicity, immunotoxicity, metallic effects, neurotoxicity, and so on. And basically what these endpoints are is a collection of the endpoints that various European agencies consider when they complete hazard assessments of various chemicals in food and also in industrial chemicals. They're broadly grouped, these endpoints, into chronic effects on the top right of the radar plot. In the bottom of the radar plot, we have all the effects that are associated with systemic toxicity. We have organ-specific toxicity. And then finally, we have in the top left, we have mechanisms of toxicity. And all of these health effects will and are considered in one way or the other when the authorities complete hazard assessments of a given chemical. And that's also why we think that they're most appropriate to have in this radar plot, because we really want the framework to be regulatorily relevant. Another key aspect of the framework that is worth mentioning here is, of course, what do you put on the axis of such a radar plot? And we have decided to put the strength of evidence. So that basically means the strength of evidence for which, that we believe it connects, or the strength of evidence for a given polymer to be associated with a given hazardous endpoint. The strength of evidence in our framework ranges from very weak to very strong. And of course, it's quite important, how do you actually define those terms? In our suggestion is that we define these terms in the same way as the European Environment Agency has defined these different kinds of strength of evidence in their work on late lessons from early warnings. David G. has proposed various strength of evidence. He's also, as you can see in this table, provided illustrative terms, examples of use. And basically, we would say that for there to be very strong evidence that a given micro and nanoplastics is associated with a given adverse health effects, for that to be very strong, we would have to believe that there's 90 to 99% likelihood that there's an association, and then gradually take it down to very weak, where we only believe that there's 1 to 10% chance that there's an association. Of course, how you also interpret these quantitative values is subject to or subjective, but it's important that you reflect on it and that you describe how you interpret these terms when you apply the framework. And here is a suggestion or some inspiration on how this could be done. When you are to assign the strength of evidence in our framework or in our evaluation scheme, we want you to use a weight of evidence approach, or we suggest to use a weight of evidence approach. A weight of evidence approach is broadly defined as considering all the information that's available, the pros and cons and quality of this information, and then trying to come to a mosaic of conclusions about what does this evidence actually show. And it can be really challenging to provide absolute guidance on how to compare one study that has some limitations and indicates a certain level of effects versus another study that is completely has other kinds of limitations and shows another set of adverse effects. But there are examples in the literature that one can use as inspiration. There's work done on chemicals, and the European Chemical Agency or the German competent authorities on chemicals, they have also completed a hazard or assessment and risk assessment of bisphenol A using a weight of evidence approach. And I think, or we think, that in combination of these examples, one can actually see how to actually go about using a weight of evidence approach, which then eventually will enable you to assign a strength of evidence that we can then put into the radar plot. One key aspect of using a weight of evidence approach is that one has to assess the reliability and relevance of the studies that have been identified as meaningful to include or maybe include in the actual hazard assessment. And there's different tools and different methods available to do this kind of assessment. Normally, registrants for industrial chemicals in Europe, they use Klimisch scores. There's also a CIRAP system that has been developed as an alternative to Klimisch. And when it comes to nanomaterials in specific, which might be relevant to nanoplastics, there's also a framework called NanoCred that could be used. Each of these tools has pros and cons, and in general, it still has to be explored whether there are plastic-specific considerations that will have to be taken into account when assessing the reliability and relevance of various studies on nano and microplastics. Rules of thumb when it comes to weight of evidence, in general, we would say that in vivo and in vitro data outweighs QSAR and computational methods. Biomonitoring, human biomonitoring and epidemiological data carries the highest weight of evidence. In vivo data usually outweighs in vitro data because they mimic human response better. This is generally the case, but it can be that there are specific situations where it's not the case. And therefore, of course, when you do the actual weight of evidence evaluation, you have to explain your reasoning, you provide scientific justification for why you weighed the different studies as you did, and then explain and justify the conclusions that you derived from your analysis. Something that's especially important to mention also in regards to our project, PlasticHeal, is that results from in vitro new approach methodologies, they outweigh results, or typically outweigh results from acellular assays or computational models. And that's also important to remember in regards to the whole shift towards moving away from full animal studies, that we want to make sure that in vitro studies, they do carry more and more weight in our weight of evidence approach over time. What I've said here about using a weight of evidence approach, that's basically in line with the guidance that the European Chemical Agency and also the European Food Safety Agency has provided on how to use a weight of evidence approach. So to summarize a little bit, when it comes to the plastic risk categorization framework that we're presenting here, there's 5 overall steps that you need to complete before you have the final evaluation. So first, it's important that you define the scope of the plastic risk cat evaluation, and then gather all the relevant information that you possibly can get access to. In our case, it's primarily the scientific literature that has been published and peer-reviewed. Then the third step is to evaluate the reliability and relevance of this information. The fourth step is to use a weight of evidence approach to assign strength of evidence for each endpoint. And once you've completed that task, you're now ready to fill in the actual radar plot, and then you have the plastic risk cat evaluation for whatever given polymer that was subject to the evaluation. The final minutes of my talk, I would like to spend on the further developments that we have planned in the project and that we will strive to complete before the end of the project. First of all, we would really like to, and we will, develop examples and on how to use the framework. We have decided to begin with polystyrene, PET, lifelike PET, and we're already in the process of applying the framework to all the data that we have generated in PlasticHeal, in CUSP, and also what's generally available in the literature. Here, when we're filling in the radar plot, when we're assessing the strength of evidence using a weight of evidence approach, we're drawing heavily on the different work packages in PlasticHeal and the experts that we have associated with the project. And you'll hear from some of them in the coming presentations this morning. We'll also not only rely on the publications and the work done in PlasticHeal, but we'll also reach out to the other CUSP projects, as well as perform standard state-of-the-art literature searches in the scientific literature. An area that we're also working a lot on is tools for assessing reliability and relevance of massive, in quotation mark, number of studies. As you saw in the first slide that I had, or maybe the second slide, is that we've seen a dramatic increase in the number of publications that have come out over the last couple of years or three. And there's just no way that we can manage to assess the reliability and relevance of 7,000 studies a year. So what we're looking into is, first of all, like, do existing schemes work well when it comes to micro and nanoplastics? And is there a way that we can use AI to actually automatically assess scores? And we've done work using, for instance, a framework with assigning Klimesh score and also a nanocred to a whole range of studies. And here the systems actually work quite well, and that's promising. So we'll further work on these aspects. We also need to have another look at whether the different criteria that, for instance, nanocred uses, whether they take nanospecific aspects into account. And we might have to tweak some of the questions that we ask so that we ensure that the evaluation we do is not only nanomaterial relevant, but also plastic relevant. Something that we're also discussing and that we decided to do, but we haven't quite wrapped our head around yet, is how to include the exposure component to the risks of plastics and nanomicroplastics, because so far what I have presented only relates to the hazards of nano and microplastics, but we know that the risks is a function of both hazards and exposure, and we would really like to also include an exposure component. We've decided that we will develop a radar plot similar to the one that we have on hazards, but we're still discussing like what the category labels should be, what should the axis look like, and how do we use a weight of evidence approach when it comes to exposure studies. And finally, we want to and aim to develop a GPT that can assist users in creating their own plastic risk cat evaluations. And I've what you see here on the right hand side is how it looks. And basically the idea is that the user can upload their own studies to the GPT, and then the GPT will automatically generate the radar plot for you, and also provide all the justification that you need for the various strength of evidence that have been assigned to the different endpoints. And of course, you can see as also noted here in the bottom of the slide on the right, like ChatGPT makes mistakes, and it's very important that we understand what kind of mistakes it's making before we want to launch such a GPT. But it's a very promising endeavor, I think. So lots of opportunities there to use GPT to auto-generate and make our lives a lot easier when it comes to assessing all the work that is being published every year. That was my final slide. So with this, I would like to thank you very much for your attention, and I'm happy to take any questions that you might have. Thank you.
Thank you, Steffen. Well, we now have 5 minutes for technical questions related to this presentation, of course. Please feel free to type your questions on the chat, and we'll try to address as many as possible. I will try to open the whiteboard as well. I believe, Steffen, that we have a question from the chat from Blanca.
Yes, I was just trying to get access to the whiteboard, so sorry for.
It's okay.
Yeah.
We are new at this as well.
Yes.
We just thought that it was a good way to interact with the attendants.
Yes.
So everybody, please feel free to jump in and write.
So the question is how to deal with missing information. And I think in general, like if there's missing information, then it would be either classified as weak or very weak in the radar plot. And you can also just put it all the way into the middle of the radar plot, so to speak, because then you're indicating that there's no information on this endpoint. Okay, great. So that made sense. It's important to maybe also explain that we will have time later today to discuss the framework in detail once the different experts of the different health effects have also presented. Yes, so there's a question about what's your plan to deal with AI-based hallucinations, and our plan is to work with the GPT that we're developing to ensure that it doesn't hallucinate. We've discovered recently that like basic sentences such as do not lie, do not make up information, read the whole article or articles before you complete your evaluation. Those kinds of sentence in the configuration of the AI, they actually limit the number of hallucinations that the AI or the GPT makes. So, but otherwise, it's a very good point.
Bettina, I don't know if it's only me, but I cannot access the whiteboard. I wonder if the rest is able to see it.
You should be able to, but we can still.
I had the same issue, but somehow if I ignore all these messages and click them away, then I all of a sudden can access. So I do not fully understand how So it might be that more people indeed have some access issues.
Okay, I hope that everybody can access the chat. I will try to see if something's going on. We have some coffee break planned before the detailed sessions about the endpoints used for the framework. So everybody that's able to access the chat and wants to ask, please use it, and also the whiteboard. And I will try to contact as much people as possible to see if they are able to do this. This is a new tool also for us, so we're just testing it and using it today. In the meantime, I think that we have another comment on the chat, and we still have like a couple of more minutes for questions.
Yes. So I see that a strategy for teaching the AI, what we've seen so far, for instance, is that we can ask it to evaluate a given study, whether it follows, whether the study or the authors used standardized methods to in their assessments. And the AI will pretty much always respond correctly in that analysis. So it's very good at identifying whether OECD or ISO standards have been used. So that's positive. But if we then ask it which standard it looked into, whether it had been used, it's at least sometimes it's the incorrect standard. So it's explaining that it's a standardized method that was used in the study, and then it's referring to a standard that has nothing to do with the subject of the study. So it might be that it's a human health study and is referring back to a standard on how to do ecotoxicity. And that's a little bit of a weird situation. So it might be that we will have to go in and let it know what kind of standards is it that we mean when we ask the question of whether a standardized procedure has been followed. So it's in that way, it's the use of AI is also challenging us to be much more clear on what is it actually that we mean when we say something, and what is like, how do we actually understand the words that we use. Yes. So how do we foresee the integration of hazards and exposure? And I think at the moment we haven't gotten around to it yet, but I think at the moment we'll keep, I think, the two radar plots next to each other, and we don't necessarily envision having a combination of the two, just like PEG-PNEC, for instance. But it's something that we'll have to think hard about, whether it's possible to actually combine the two. So there's a comment on read-across strategies for nanomaterials that may be applicable to micro nanoplastics, but reliable characterization methods for these materials are currently limiting this option. Characterization is a very important issue, and the way that we think about it in the approach that we're suggesting is basically that it has to be part of the weight of evidence evaluation that is being completed in the as part of the evaluation. When you look at the quality of a study, the first thing that we would normally recommend that you do is to look at, like, how were the material characterized, and how sure are we that what was used in the test is actually a reflection of what is proclaimed to have been tested using various characterization methods. We don't see necessarily that characterization of nanomicroplastics has to be part of the the radar plot or the axis of the radar plot in and of itself, but it is a discussion that we had when we were developing the framework. I think that was all the technical questions related to the presentation. I look very much forward to having a roundtable discussion later this morning about the pros and cons of the tool and how you could see that we can actually use it in practice, and also whether you feel comfortable with actually assigning very weak to very strong labels on the level of evidence that we have.
Okay. Oh, Juliana has a last question, so I believe.
Yes, the question is, how is this different from the risk assessment framework tool? And I must admit, I'm not 100% familiar with the risk assessment framework tools. So as far as I understand, it's based very much on NAMs and inhalation exposure, but I might be wrong. I know for sure that it's not similar to all the other projects in CUSP. So Aura are focused very much on creating various flowcharts, a roadmap for looking at exposure for fetuses and the unborn child. Imtox is looking very much at exposure assessment. Nanoplastic Fate is looking very much at developing a multi-criteria decision strategy. So I think overall, as far as I know, the different projects have developed different novel risk evaluation frameworks that could be used for nano and microplastics. And the discussions we've had with the other projects, it seems that we're actually supplementing each other very well. And at the moment, we've decided to all study and apply our frameworks on polystyrene, and then bring all the knowledge that we've generated in the different work packages of all the projects into play, and help each other apply all of our frameworks so that we collectively have a really nice understanding of the pros and cons of all the frameworks and how they can supplement each other along the life cycle of nano and microplastics.
Okay, so we have planned for a 10 minutes break, but it's up to all of us what we want to do. If we want to have 5 more minutes for a coffee break, or do we want to continue? I don't know, Alba, Steffen, what do you want to do?
Virginia, I'm ready, but I have to deal with some noise here in the next room. So I need at least 5 minutes to solve the issue.
Okay, so let's do this. I will try to solve some access problem from people that cannot access the chat and the whiteboard, and we can use these 5 minutes for everybody to go grab a coffee or something to eat fast, and I will leave the whiteboard open for all of you that want to ask, and even you can you still can comment on the chat if you want to. But we will have a 5 minutes, and we will go back live once Alba is back.
Okay, I'm ready to present, if that's okay, Virginia.
Yes, perfect, Alba.
Okay. So carcinogenicity and mutagenicity is one of the categories of the radar plot that Steffen just introduced to all of you. So the idea of my presentation is to give you an overview of what is the evidence that we have collected in the project in PlasticHeal regarding this endpoint. The first thing when considering carcinogenicity and mutagenicity is that those are effects that typically happen after long-term exposure or under chronic scenarios of exposure. So let's see what's the evidence regarding these and micro nanoplastics. So we know that we are chronically exposed through ingestion and inhalation because there's micro nanoplastics that have been found in the air and also in the different foods after entering the food chain. So this is during the whole life period. And for those micro nanoplastics that are smaller, let's say less than 1 micron, less than maybe up to 3 microns, they have the possibility to cross these primary barriers after ingestion and inhalation. And actually, this has been demonstrated, this translocation from the primary organ to the blood has been demonstrated by many authors, including us inside the consortium. We have worked with complex barrier in vitro models, both from the gastrointestinal tract, as the one that you see here, and the respiratory tract that you see here. Those are complex models, cells that are tightly organized as an epithelium, and where we can explore how these micro nanoplastics enter. You see them on green here inside the cells and then translocate through the system. This has been demonstrated not only in vitro but also in vivo, again by different authors and by us, using different in vivo models. One is the alternative Drosophila fly, but we have also generated evidence in mammals, mostly in mice. So you see here a picture on how we can track in the Drosophila in this case, as an example, the plastic approaching to the gastrointestinal epithelium, crossing it, and reaching what is the equivalent of the blood in the fly, the equivalent to human blood. And so if there is this translocation, we assume that humans can be exposed, the primary and secondary organs can be exposed long term. And actually, there's a bunch of literature out there stating that micro nanoplastic can be found in the many different human organs. We can complement this evidence with our own PlasticHeal evidence. Since we have found micro nanoplastics in the blood and exhaled breath condensate of our case studies, which are people exposed at the workplace. Be aware that we have, or consider also that we have found micro nanoplastic in the controls, not only in the supposedly exposed population. And we have found different polymer types and in the nano and the micro range. We can argue and discuss about what's the quantification of all the exposure, but it's quite clear, at least qualitatively, that micro nanoplastic reach different human organs. And then we have clear evidence that micro nanoplastic have the chance to bioaccumulate there in primary and secondary organs and induce long-term effects. Let me see if I can access the next slide. Excuse me. Okay, so after talking about the exposure, let's see what we know about the effects, carcinogenicity and mutagenicity effects. If we look closer to the carcinogenic process, not in very much detail, but just as an overview, we can expect that when the micro nanoplastic particles enter into the organ, there's a pro-inflammatory response that leads to a recruitment of inflammatory cells into the tissue. This typically generates a situation with oxidative stress that can eventually cause genotoxic injury in the cells inside this tissue. If a mutation in these cells directly, that could lead to transformation of the cells. Under a scenario of chronicity where this is sustained on time, we can also have chronic inflammation leading to fibrosis and leading to changes in the transcription of different pathways that are related to the carcinogenesis process. This means that if we understand the carcinogenesis process as this whole, we can both measure directly the event of acquiring the cancer-like phenotype, or we can measure each one of these points as a surrogate biomarker of the carcinogenesis process, being genotoxicity the gold standard as a surrogate for carcinogenicity, but also inflammation plays an important role here. So taking this into consideration, we conducted a systematic review and we analyzed the literature. We finally end up with 26 articles after applying the inclusion-exclusion criteria. And we realized that there is no direct evidence of cell transformation or carcinogenicity from micronanoplastic, and that we have to rely on surrogate biomarkers only. If we take into account these surrogate biomarkers, we found that there's evidence of this being genotoxic mutagenic, also causing fibrosis, and also able to trigger sustained inflammation. This taking into account that all the studies considered also had information about the accumulation and exposure in the target organ or cell that was analyzed. You have here a summary of one of the tables of this exercise. This is systematic review. And you see here that there's found these surrogate biomarkers. Here, inflammation, fibrosis, genotoxicity. This is only in vivo and with different regimes of exposure. Of course, this comes with limitations because the literature is limited. It's only a bunch of articles. And as you see here, most of the material that has been used for such studies is only one polymer type, which is polystyrene. So there's evidence that there's a potential there for being carcinogenic, but still limited evidence. How can PlasticHeal complement this information that we have in literature? We have worked, or I have summarized here the information of two different polymer types we work with in the project. One is polystyrene of two different sizes, 50 nanometers and 200 nanometers, and also real-life PET that we have obtained from the degradation of plastic bottles. When we assessed the genotoxic potential of these 2 or 3 particles, we see that in general polystyrene shows as more genotoxic. We have found genotoxicity in white blood cells from peripheral white blood cells from blood from donors. We have also found some positive genotoxicity in long-term exposed cells, transformed cells. They are regular cells that we use in the lab. Positive in Drosophila and also in mice. Of course, we need to consider that there's a lot of influence of the experimental settings on this, and that each one of these cells or animals have been exposed a little bit differently. So we need to account for that. As I said, PET shows us less genotoxic and more triggering of inflammation. And you have here, just as an illustration, you have here a graph with the same experimental settings. This is mice after having inhaled the particles. And as you see here in the lung, this is PET, which shows less genotoxic than the 2 sizes of polystyrene. But on the contrary, in the liver, in a secondary organ, PET also shows genotoxic. So we need to manage the information here carefully, taking into account all the different experimental settings. But in the end, there's a support from our side regarding these surrogate biomarkers that we found in the literature. What happens when it comes to measure carcinogenicity directly? We tried to avoid from using these surrogates and directly measure carcinogenicity. And we have bet for two different systems in PlasticHeal. The first is the Bus cell transforming assay that's recognized by the OECD as a reference in vitro method for this. And the other one is what we call the long-term exposure system, which consists in exposure a given cell during weeks or months to low doses of the micro nanoplastics, and then assess through time different features of different hallmarks of carcinogenicity, from proliferation rate, morphology changes, secretome changes, to the ability of the cell to grow independently of the substrate, the migration or the invasion potential, or the ability to generate tumor spheres. Together as a whole, this battery gives us the information on whether at a given point the cell has been transformed by the micro nanoplastic chronic exposure. So let's see what we found for this nanopolystyrene and nano-PET that we were discussing previously. The VAS 42 cell transformation assay for nanopolystyrene 50 nanometers shows negative results. You have to look at these columns here. These are different doses of the polystyrene. And as you see, there's no one statistically significant, meaning that this particle was unable to show the initiation of the tumoral phenotype. Neither for the promotion. As you see here, the different doses, these columns, and none of them is significant. So polystyrene shows negative for this assay, and we find the same if we use our long-term exposed system that I described to you, using normal cells, pulmonary cells of epithelial origin. But let's look at what happens if we consider all the conditions and not epithelial cells from the pulmonary organ. You see here this is the result from the nanopolystyrene analyzed in cells of stem origin. And as you see here in these graphs, you see this is polystyrene, which is in between the negative and the positive controls for the ability to grow independently of anchorage. This is polystyrene in between the negative and the positive control, again, for the invasion capacity. You see the same here in the form of bars. So it looks like there's a tendency in the long-term exposed stem cells to show some features of cancer hallmarks. And when we combine this information with the molecular alterations that we find after omics analysis, somehow we confirm that there's a transform phenotype in these circumstances. When it comes with a situation or a setting of co-exposure, this means polystyrene with another genotoxic of carcinogenic compound, again, we see positive results for the cell to be able to grow independently of anchorage, migration capacity, and invasion capacity, meaning that at the functional level, polystyrene is exacerbating the oncogenic phenotype that's induced for this co-exposed agent. Let's see what happens now, what we have found with the real-life nano-PET obtained from the degradation of the plastic bottles. If we look at the VAS cell transforming assay, and we again look for the initiation and the promotion side of the assay, you see here that there's no statistical significance, but there is statistical significance at the higher doses of nano-PET for the promotion ability. So nano-PET gives positive results at high doses in the VAS cell transforming assay. When we move to our long-term exposure system with the normal regular pulmonary cells of epithelial origin, we found that after 30 weeks of exposure, we found significant increases in the invasion and ability to grow independent of anchorage. Also, this oncogenic phenotype is again rapidly exacerbated if we co-exposed with a known genotoxic or carcinogenic agent such as the tobacco smoke condensate. So we see the ability of nano-PET alone to induce transformation and also the synergistic effect with these co-exposures. It's interesting that when we look at the transcriptome of these long-term transformed cells, we see deep alterations that are consistent with the profile of lung cancer. You see here all these different deregulated genes after 30 weeks of exposure. And when we look at the pathways where these genes are involved in, many of them are related to cancer. Actually, you see here some typical genes associated with lung cancer, and you see how significantly those are deregulated after chronic exposure to nano-PET. Here you can see it actually more clear. This is a pathway that's involved in lung cancer. And you see in each one of the boxes, it's a gene in the whole pathways that controls this cancer. And then you see that all what's in color is deregulated due to the nano-PET exposure. And interestingly, this effect that we are seeing of the transcriptome, it's depending on time. If you look at this graph, you see here this flat line is the number of, or the impact on the transcriptome of nano-PET at 24 hours, which is no impact at all. There's no response or very no response. Then you see here, this area here, darker means 15 weeks of exposure. So there's already some response. And this is what you see after 30 weeks of exposure, meaning that the response on the transcriptome is very clearly time dependent. And here is more or less the same thing, but looked in another way. These are genes related to the epithelial to mesenchymal transition, which is connected to carcinogenicity process, and genes that are involved in lung cancer. And you see this column is the acute exposure, 24 hours, 15 weeks, and 30 weeks. And you see that for a lot of these genes that are involved in very important processes for carcinogenesis, there's a time dependency, there's a progression of the regulation with time. So we have to summarize all the results that I've shown you. We have here the nanopolystyrene and the real-life nano-PET. There is enough evidence for both of them to consider that this can be accumulating in some organs, and on the long term, this can have significant effects. There's evidence of genotoxicity more for polystyrene than nano-PET, but on the contrary of this genotoxicity for carcinogenicity, PET is the one showing positive for all the experimental settings that we have considered. This is the OECD VAS cell transforming assay, the long-term system with the normal epithelial cells, the context with co-exposures, and the particular context of stem cells. And nanopolystyrene is very responsive in a scenario of co-exposure and in certain cell types like stem cells, but is negative in the regular normal epithelium tissue and the VAS cell transforming assay. So we consider the weight of evidence as moderate because there's reasonable grounds of concern, but not sufficient scientific evidence. Most of the ones that are directly measured carcinogenicity are only based on in vivo systems. So we need to collect more evidence in the future to be able to understand whether this is significant for in the context of real or a real human exposure. So with that, I would like to finish. I would like to thank the fantastic PlasticHeal team that made possible all these results. Also the cluster that work coordinately with us in trying to understand the health impacts of micro nanoplastics. And thank you for your attention. And if you have any questions, I'll be happy to answer now, if they are technical, or later in the roundtable.
Thank you, Alba. We will have, again, 5 minutes for technical questions. And please use the chat. I understand that some people are having issues with it. I'm trying to solve it. And I will activate again the whiteboard so that you can play a little more. Alba, are you able to read the questions from the chat while I do this?
Yeah, I'm going to stop sharing.
Okay.
It says that what would advise for the next steps to continue gathering evidence on carcinogenicity. So what I would say is that we need to, for sure, take into account the mixture situation or the co-exposures, because derived from our investigations, it is quite clear that this scenario is of potential concern. When including or considering these mixtures of this co-exposure, it's quite important that we take into account additives that can come with the plastics, so we can really understand whether there's a combination of the chemical and the particle in giving a carcinogenic or genotoxic response. And of course, at some point, it would be needed some in vivo evidence. It's important also to take to consider different doses of the same compound and see what is the dose response. If we have a threshold there, this is something that we have not done yet. So I would say that there's a lot to do still. But for sure, mixtures and additives in the equation, I think it's quite an important thing to take into account. It looks like there are no more questions. Going back to the comment of Steffen on whether considering exposure and effects separately, notice here that I've tried to make a kind of an holistic view of taking into account both, no? Because at some point they are connected. If you don't have enough evidence of exposure supporting that a given effect would occur, no, it's an element that also needs to be included in the equation. Just for you to reflect on this.
Okay, it appears that people are starting to play with the whiteboard, so that you know there are more experts from the PlasticHeal team here that are also using the whiteboard, and I see some of them answering some of the questions, or at least the first one that is showing. We also have a couple more of minutes. I will leave it 2 more minutes in the whiteboard active, and then we can go. If there are no more questions, we can go to the next presentation. So I leave you 2 more minutes to play with it.
Alba, I have a question. I'm just going to open my mic and ask it the old-fashioned way, I guess. You you did a review of the literature, and you identified, I think it was a little bit about, was it 125 studies, but only 26 or 25 of them were considered relevant. I was just wondering, like, how come so many of them were irrelevant? Do you remember?
Because yeah, well, if I'm not wrong, this is because we decided to consider only the human cells or mammary cells, like models. We included only genotoxicity with models that can be meaningful for human health, and we left aside a lot of information that was gathered under a more ecotoxicological point of view. Because there are a lot more information on those models. But we wanted to, restrict the field and focus more on human health.
Thank you.
Okay, I will stop sharing the whiteboard now, and we can, if it's okay with you, we can go on with the next presentation.
There is, Virginia, there's a question in the whiteboard.
Okay, go ahead.
It says, did you use a 3D cellular model or a cancer cell line? I guess that you mean for the long-term experiments. For those experiments, we have used different cell types. It was not 3D cellular model. It was a regular cell lines that grow like in culture. But we have used normal cells, cancer cells, stem cells, epithelial cells. We have used many different cell types for the assessment. Okay, that's it. We can move on, Virginia.
Okay, so maybe it's time for welcoming Diego to explain a little bit more about the immunotoxicology.
Good morning, everybody. I hope you can see my presentation. And I'm going to give you a talk about immunotoxicology, specifically how micronanoplastics affect the immune system. Let me say that we are from IMIB in Murcia, and we are part of Work Package 5 within the PlasticHeal project, in which there are some other research institutes, so we are a big work package. So let me start with a little introduction about, as you know, a lot of plastics are in our lives. These plastics suffer some kind of degradation, can be physical, can be chemical, and this degradation makes the size of these plastics to be smaller. And this can be incorporated into the human body. As Alba has said before, the principal ways to incorporate these micronanoplastics are when we breathe, we can breathe them, we can also eat them, and they can be accumulated in, let's say, the first barriers are the lungs or the gastrointestinal barrier, in which we have some kind of immune cells, such as the dendritic cell of macrophages, that, as you know, they can accumulate micronanoplastics. In here, in the picture, you can see human macrophages, where in blue we have the nucleus, and in red we have the actin, and in green we have the fluorescent polystyrene particles. So, these immune cells are the responsible to produce, let's say, a fast response as a local inflammation, and if this local inflammation is not solved, we can produce, these cells can produce a long-term inflammation we call systemic inflammation that can affect all the systems and is characterized by a subclinical low-grade inflammation where the adaptive immunity is involved. So yesterday, making a quick search in the PubMed, I typed microplastic and inflammation, and I found around 300 papers where most of them talk about a specific disease and the effect of micronanoplastics in this specific disease, for example, in the central nervous system, about cardiotoxicity, about a specific disease in the gastrointestinal, about the kidney function and respiratory disease. And most of them use the mouse model the mouse model is used more than the human model. And if we go deeper and we type micronanoplastic and inflammation, I found 5 results where only 2 were related with micronanoplastics, and 1 was a review, and other was a study using THP1 cells, that is an immortalized monocytic human cell line. But of course, if you go deeper and type some specific words about inflammation, you can find several studies. In here, I just have put a few samples where they use the mouse model to study the effect of polystyrene in the gastrointestinal disease. As Alba has said, most of them use polystyrene, and always the same plastic. They also use other models at the chicken to study the cardiotoxicity and the kidney function using the mouse model. And of course, the human model is also present using primary human monocytes and dendritic cells, and also using polystyrene that had been aminated. That, of course, as you know, this is not a true-to-life plastic. So we are talking about inflammation, but what is the inflammation? Let me introduce you the inflammation, because this is one of the platform we are working with. The inflammation is an immune sensor acetylsolic 1 that is formed by a sensor protein, for example, the NLRP3, one adductor protein is called ASC, and one effector protease that is the procaspase 1. After a specific stimuli, this protein can bind by homotypic interaction, forming what we call the inflammation. Once the inflammation is formed, caspase 1 can be activated. And this effector activated protein can cleave different proteins, including pro-inflammatory cytokines such as interleukin-1 beta and interleukin-18. Because of the activation of the inflammation, some pores can be formed in the cell membrane. Through these pores, the active interleukin-1 beta or interleukin-18, can be released to the cellular media. And this can produce a low-grade inflammation. So from now, you will see that we are going to measure the release of this pro-inflammatory cytokine, the interleukin-1 beta, as hallmark of inflammation activation. Specifically, we are going to work with the NLRP3 inflammation. This is one of the most important inflammations. It has been widely studied and has been reported that can produce some sterile inflammation in response to nanoparticles such as silica or asbestos, and can induce some kind of low-level chronic subclinical inflammation. So IMIB have studied how the micronanoparticles of plastic can affect to the inflammation, the NLRP3. How micronanoplastic can affect the inflammation response? Well, it depends on when the plastic is present. So for this, we have made 2 approaches. One approach is to expose the nanoplastics after the infection. So we work with some immune cells that had been infected, and then we add the nanoplastics to see if they can produce an inflammatory response. And a second approach, we have performed an exposure of nanoplastics before the infection. So we have put the different nanoplastics in the immune cells, and then we have performed an infection or a classical inflammation activation to see what happens to the inflammatory response. Now I'm going to present you some results we have in this regard. For example, regarding the first approach, when the exposure of nanoplastics is after the infection, we have worked with different kind of micronanoplastics, in which some of them are true to life. And in here, for example, using bone marrow derived macrophages from mice, wild-type mice, we have infect these macrophages, and then we have added different micronanoplastic and check for the release of interleukin-1 beta as hallmark of inflammation activation, as I told you before. In here, you can see how polystyrene and PE are able to increase the release of interleukin-1 beta. They are able to activate the NLRP3 inflammation. Regarding the others, PAT, PLA, PBC, PNA, they were not able to induce any release of interleukin-1 beta. They were not able to activate the inflammation. It's important to say that although we use low concentrations to see interleukin-1 beta release when we use PS and PE, we use higher concentration for the others to see if we were able to see any kind of interleukin-1 beta release, and we were not able. If we move to the human body, to the human model, we also check for interleukin-1 beta release in PBNCs isolated for the samples of whole blood, and we see how PS is able to release interleukin-1 beta. And we also check for the specificity of NLRP3 activation using a specific inhibitor of the NLRP3 that is called MCC 950 that were able to inhibit the release of interleukin-1 beta due to the presence of PS. Because we have different sizes of polystyrene, we wanted to check if the size was important to obtain inflammation activation. Using the same approach, using bone marrow derived macrophages from mice, we infected them, and then we add the different polystyrene with the different sizes and collect the supernatant to measure the interleukin-1 beta release. In here, you can see how all of them were able to produce the release of interleukin-1 beta. They were able to activate the NLRP3 inflammation, but it's important to say that the smaller the particle, the higher number of particles you needed to activate the NLRP3 inflammation. Now, if we move to the second approach, to add the different micronanoplastics before the activation. In here, I'm going to talk you about the human model, specifically using PBNCs. We isolated the PBNCs, we add the different micronanoplastics, and then we perform a specific NLRP3 activation. And you can see how when we measure the percentage of interleukin-1 beta release, in here you have the normal activation, the 100% more or less, with no plastics. You can see how the presence of PS and PAT before the NLRP3 activation affected the NLRP3 activation compared with the normal response, while the others, PLA, PBC, PNA, and PE, were not able to inhibit this percentage of this release of interleukin-1 beta release, were not able to inhibit the activation of NLRP3 inflammation. Then we wanted to see if there were any differences between gender. So as you can see here, the presence of PAs affect in the same way to men and women, while the presence of PAT and PE, it seems that men are more susceptible to the presence of these microplastics than women. I would like to say that we obtained the same result using isolated monocytes and human macrophages, but in the case of human macrophages, we also see that PE were able to produce an inhibition in the NLRP3 inflammasome. Now, moving to the PS of different sizes to check the action of the size using the same approach, you can see here how PS from 15 to more or less 100 nanometer of size were able to inhibit, to decrease the release of interleukin-1 beta, so they are able to inhibit the NLRP3 inflammasome activation, while the larger one, PS200 and PS400, do not. And when we move to see the differences between gender, we saw that we didn't find differences between men and women. Of course, we obtained the same results using isolated monocytes and macrophages. And just to finish, we also wanted to know what happened with the adaptive response. For this, one study we have performed was to study the internalization of PS, and PLA in T cells, in total T cells. As you can see here, PS can be internalized by T cells, and after activation, this internalization continues if you compare with the T cells with no plastics. And what happens to the PLA? PLA can be also internalized, but this internalization increases when these T cells have been activated. This is a quite novel result because, as far as I know, there is no any report that any study that report this. And then we move to see what happens to the immunoglobulin A using B cells. As you know, immunoglobulin A is a classical actor to check the adaptive immune response, and also has an important role in the mucosal immunity, specifically in the epithelial cells that, as you know, are the first barrier that the micronanoplastics are going to find. We check what happened to the release of immunoglobulin A when PS and PLA were present. As you know, when PS and PLA are present, the percentage of immunoglobulin A is decreased. So the presence of these plastics can affect also the adaptive response. Just to conclude, I would like to tell you the conclusions. We have numbered the different conclusions, and we also have classified the weight of evidence based in our results in strong, moderate, and weak. So, as conclusion, we can say that PS and PE after infection can activate the NLRP3 inflammasome. The larger of the PS particle, the fewer number of particles are required to activate the NLRP3 inflammasome. PS and PAT before infection inhibit the NLRP3 activation. Males appear to be more susceptible to the presence of PAT and PE in NLRP3 inhibition than females. PS particle from 15 to 100 nanometers before infection inhibit the NLRP3 activation, while the larger ones do not. PS of different sizes before infection have the same effect on men and women. T cells uptake PS and PLA. The uptake of PLA is increased when T cells are activated. To finish, PS and PLA before B cell activation decrease the amount of immunoglobulin A. As you know, most of the conclusion we have classified them with a strong evidence. The moderate ones are related with the difference by gender, because we need to increase the number of samples in men and women to be sure about this statement. And then we have a week one about the PAs and PLA before B cell activation, about the release of immunoglobulin A, because we only have performed this assay twice, and we need to increase the number of assay to increase the weight of evidence. And that's all. Thank you for your attention.
Thanks a lot, Diego. You already have a couple of questions and comments on the chat, and now we have the playtime with the questions and answers. And before I activate the whiteboard, I will also launch some questions that Diego wants to ask you as well. So you should be able to see a poll from the teams. I hope not to have a lot of technical issues with this one.
We see it.
Okay, so you should be able to, on your own rhythm, answer. These are yes and no questions, so it should be quick. And while you do that, Diego maybe can also check the chat.
Yes, I'm reading a question that have you compared the observed effect of nanoplastic with other non-regulators, non-regulators of the NLRP3 pathway? Yes, in this regard, we always put a control that is silica as a positive particle that activate the NLRP3 inflammation, just to have a particle that is not a microplastic, but is a particle that we know that can activate the NLRP3 inflammation. But is the number of samples significant regarding distinction of men and women? No, that's why we think we need to increase the number of samples in this regard. We have to increase the number of men and women to be sure about to make a significant distinction between men and women.
But Diego, this is something important to highlight, no? That I think that we have collected evidence, not only the one that you presented, but the one also with single-cell RNA-seq analysis and others that points towards a potential difference or a differential impact influenced by the gender. So it is something to take into account in future work. Because it's true that probably we will need to increase the number of subjects, but there's all the information that we manage inside the consortium points towards a potential difference.
When you say that more small particles are needed to induce interleukin-1 beta release than larger ones, does it correlate in the end to the total mass of internal plastic? This is something we are working with. We are trying to count or to have a number of different size of plastics in the different immune cells to be sure about that. Carcinogenicity. Do you think that this is something I have wrote?
Yes, you're probably looking at your own questions, Diego.
Yeah, the last one was yes. How can I see the poll?
If you go to the chat, you can click on
I'm
view
clicking,
poll,
but I'm not seeing it.
and if you answer,
Oops.
once you give all the answers, you are able to see the results.
Hi, Diego. Have you done in vivo studies as well? No, we haven't done that. I think Cyrill is going to talk about this. I think Cyrill have this model, a proper model to do this.
Well, I can say already that we are planning to actually focus on inflammasome at the beginning of the year. So we will have a small study that will look specifically at the activation of inflammasome in vivo.
Great. That's a good question. For the gender-specific effect, do you think it is related to uptake of particles or potentially hormone-related pathway effects from plastic-associated chemical or other reasons? That's something we have to explore if we see a clear difference. So right now, I don't know what to say about that. I don't know why women can be more susceptible to, or sorry, men, to PAT than women. I really don't know. The whiteboard, okay. Okay.
Okay, we have 5 more minutes for questions. Maybe it will be useful for people commenting on the whiteboard to put their names, probably. We have a comment which says, yes, several of use data on that. Obviously, the uptake of the is ice dependent and can be inhibited by specific endocritic inhibitors. I'm not sure if...
Yeah, that's my comment, but I don't know how to personalize it, I would say.
I didn't see that comment.
That is a question, and I commented on that.
Maybe just jump in, Hans.
Yeah, but I cannot. I see it. I posted it anonymously, so I'm not sure how I can put my name to that.
Have you found data on the cellular uptake mechanism? This is a question for me.
For everybody, I guess, for the whole consortium, I guess.
Because we are working about how different nanoparticles can be internalized. And we are working with autophagy and so on. So we have some other result about that, but we need more time to discuss that.
Okay, I see people moving their screens, but if that's okay with you, we can move on.
Okay.
And I think that there will also be technical questions for Cyrill. Afterwards, we will have the roundtables, and we will still have more time to discuss on other issues. So Cyrill, whenever you're ready.
Okay. So hopefully everything is displayed properly. Okay, so now I'm going to talk about the gastrointestinal effect, so the impact of macronanoplastic. So that's not the result only coming from here, Manchester in the UK. It's actually the gathering of all the information that we have collected over the last few years from the different institutions. So I'm going to focus only on the effect mostly, but you also need to understand that as part of that, because that's the main stream of exposure of humans, there has been also quite a lot of work on the exposure. So we have done quite few works on trying to understand how much is present in food products. So mostly juice, milk, and water as well, trying to see if the storage condition is impacted. Now, that's something that will be also coming across tomorrow when we will explain all the exposure. And that's also a key important aspect to know roughly if the dose we have used are relevant or not. So whether or not we actually predict accurately what is human exposure with what we have been trying to model. So the first part, if I can move to the next slide, but apparently there is some problem, will be about just giving you a brief overview of what is known at the moment in the literature, so where we are and where PlasticHeal is making a difference as well. So I will definitely always compare what is the exposure versus what is the effect. So as you know, yeah, exposure is due to the fact that we are overusing plastic. The waste is not well managed, and those wastes are breaking down, going everywhere in the environment, and therefore over time reaching the food chain. And at the end, the super predator, who is the humans, is getting impacted as well and contaminated. So in terms of gastrointestinal tract, that will be all food, drinking, and also inhalation for whatever is inhaled as well, but whatever is coming back from the lung via the mucociliary clearance will also get back into the gastrointestinal tract. So that means that both inhalation and ingestion are actually relevant somehow for this gastrointestinal impact. So that's why you will see that in some cases we are also considering inhalation. At least the literature has been consider inhalation as also a way to increase the amount that we are exposed to. So yeah, the literature is definitely showing that humans are getting exposed because there is evidence of presence of this macronanoplastic in food system from the food production. So that means the raw material is already contaminated. As soon as the food is processed as well, the packaging is adding some plastic. And at the end, what we have in our plate ends up also contaminated by all this accumulation of plastic. Just to highlight roughly what is estimated. So you see that there is a lot of variation depending on the study, with some high numbers, some small number, and it's all depending on where people are based. So there is one study that was actually quite nice because they were trying to do, based on some experiment, an estimate for what would be the overall ingestion over a year. So that's the estimation from 39,000 to 52,000 particles, depending on age and sex. And you can see that there is a split between male, female, adult or child, and that's all depending on behavior. And then if you add people drinking bottled water versus tap water, there is also a big difference, with the bottled water increasing dramatically the amount of particle in the food system. So overall, what it looks like is that it's all depending on behavior. So consumption habits will influence dramatically how much people are exposed to, and that will also obviously depends on where they are located as well. So that means that it's difficult to actually have a very accurate estimate of how much we are exposed to, because there is so many variables. And that's another one showing you a little bit what people have found in terms of what is found. So fiber are present quite a lot, and then there is the fragment and also some granule. And obviously seafood is one of the main components, because what the first picture was showing you is that they are exposed to this plastic pollution in the ocean. So that's where the maximum exposure is currently happening in terms of our food chain. Yeah, so that's the next one. So moving from where the food is up to the humans. So that's how people are assessing whether or not humans are exposed and how much they are exposed. The typical technique that people are using, PyroGCMS, macro-Raman, or macro-FTIR. And that's what they have found. So you see that there is a lot of different particles that can be found in stool. There is the same outcome for urine. So there is quite a lot of evidence that actually we are exposed and we are getting material in, at least. In terms of effect, so far, there is no clear defined evidence that this exposure can be linked to disease, but in one study, they have actually compared two cohorts of people, healthy ones versus people presenting IBD conditions. And what they found is that actually there is correlation between presence of a higher amount of macronanoplastic in the stool of IBD patient and the IBD severity. So that may or may not be the cause. So that's the key question. There is only correlation at this stage, and IBD may actually influence our macronanoplastic are eliminated. So if we go to people modeling what is happening to try to understand, what people are typically looking at is gut microbiota, whether or not the mucus layer is affected by the exposure to macronanoplastic, and whether or not the cells involved in the epithelial barrier are suffering from the exposure. So what has been found is that there is definitely an impact on the gut homeostasis with modification of the microbiota, alteration of the mucus, and also damage to the epithelial barrier integrity. And in terms of mechanisms, oxidative stress and inflammation, the same as what was found as culprit in nanomaterial, is also the one that seems to be involved here. And the effects have been attributed to the dose, to the size, and the time as well. But what is not found at the moment in the literature is actually what is really relevant for human, which is the long-term effect due to a chronic low-dose exposure. So now moving on to what plastic has been trying to produce, whether or not we have filled some gap in this uncertainty. So first, we actually are working mostly on nanoplastic, and for that we are comparing commercial PS nanobits used as benchmark and a new product that we consider true to life, really representative of what we are eventually getting exposed to. So most of the material, when we say PT, nano PT, is coming from a transparent plastic bottle. We also have the same, but from milk bottles. So in that case, they are opaque. So that means that there is a bit of agent to stain the material compared to a transparent plastic. And we also have experience with tea bag, with different type of polymer that have been tested and the release from the tea bag. In terms of models, what we have used from very simple cells, but also advanced model where we expose for a long period of time to see how the cells are changing, more complex model where we put all the cells that are part of the gastrointestinal barrier, and more recently also some intestinal organoid-like model where all the cells are present and they are coming from iPSCs. And then in terms of more complex, more advanced, we are using also organisms. So the fruit fly with the larvae, because the larvae is just basically just eating all the time. So that's a very good model to check gastrointestinal damage. Then we have used the mouse, and we're also going to compare all this model to what we are happening in humans with different cohorts of workers in textile facility and recycling company as well, to know if there is correlation between what we found in our models and what has been found in the humans. So let's just show you some of the results. So first, starting by single cell model with the tea bag releasing. And in that case, we wanted to know whether or not these tea bags were releasing nanoparticles and whether or not these nanoplastics were actually accumulating within the different cells. And what we found is that actually what you see here in green are those nanoplastics, and they tend to accumulate in a very cell type specific fashion. And surprisingly, they are actually accumulating in the cells that are strongly secreting mucus. So not the typical epithelial cells, but actually the ones secreting the mucus layer. So that was a bit surprising because it's not always that common. And as you can see, the cells are healthy, so there is no evidence of toxicity, but eventually some evidence of small challenge to the cells as they are behaving. Another one was, in that case, the same, but we look at polystyrene, and obviously what is typically happening within the gastrointestinal tract is that the particles are getting modified by all the digestion enzymes that are present within the gastrointestinal tract. So we wanted to know whether or not pristine versus digested polystyrene will be changing the outcome on different cell types. So in that case, focusing on 3 cell lines. And what was found is that the digested polystyrene tend to agglomerate more and also to accumulate much more protein at their surface. So that leads to much more recognition of the nanoplastic by the cells, and therefore more accumulation within the cells. And what also is interesting is that that coating and modification by the digestion enzyme led to a reduced impact in terms of effect. So just to show you here, at the top you have the effect of the cells without digestion and the effect with digestion at the bottom, and you see that at the bottom there is no effect whatsoever. So the representative more relevant model, which is at the bottom, is showing no effect from those polystyrene particles. Then the next one was, in that case, looking at exactly what is attaching at the surface of those digesting polystyrene nanoplastic. And what we found is that there is a large amount of different protein that attach. So the corona, which is this large composition and complex composition that tend to change over time, is definitely impacting the cells. So in that case, macrophages are recognizing the particles and are damaging the effect could be. And in terms of uptake, that was very much depending on what was attached at the surface of the particles. So that just to show you the range of different effects that were shown, definitely showing that there is much more increase of uptake for the digested particles. So again, if you look at more relevant system, you see that you end up with what is the reality for humans. So then if we move to one of the advanced models, so in that case, the full replication of the GIT barrier with all the different cell type, epithelial, the one secreting the mucus, and some immune cells at the bottom. And we wanted to know whether or not there will be an effect. So in that case, that was using the benchmark particles of polystyrene again, and there was no evidence of toxicity, no evidence of damage of the barrier, and the particles were able to cross without damaging and getting into the other side and reaching the immune cells. And overall, there was no evidence of oxidative stress or genotoxicity in those particular cells. That's just some of the outcome, and you see that everything is flat. The only difference is when you see here at the bottom, the increase is when actually there is dose-dependent increase of uptake, increase of the uptake. Another advanced model that was developed was the long-term impact of this chronic exposure to micro nanoplastic and nanoplastics. So in that case, polystyrene particle, where we expose for long period of time. So here, 8-week exposure to intestinal cells. So no evidence of toxicity, but evidence of increase of anti-inflammatory and anti-oxidative stress genes, so that therefore was somehow suggesting that even though the particles are not toxic, they may stress the cells, inducing some long-term damage. But at the same time, when you check whether or not that translates, so the alteration of the genes translate into event, there was no evidence of oxidative stress, and there was no evidence of DNA damage whatsoever in that particular model. Then if we move to the in vivo work, so first with the alternative to mammalian model. So as I mentioned already, one of the key aspects of this particular model is that the larvae is just constantly eating. So therefore, when you look at the gastrointestinal damage, that's very interesting, because that's the perfect model to actually just look at what is happening. And the first thing that we looked, so here I'm just summarizing 4 different papers where we look at the benchmark polystyrene, PLA nanoparticle, PET, and the PET with the coloring agent, which was titanium in that case. And what you can see, just presenting the result for the PET with all the different outcomes. So you see that for all the doses, even the smallest one, there was definitely increase for general stress, intestinal physical stress, and also dose-dependent increase for oxidative stress, and DNA repair was also stimulated. So in both cases, that was definitely suggesting that something else was happening compared to what we have seen previously in vitro. So therefore, we started to look at the oxidative stress and the activation of ROS. And what you can see here for all the different material tested is that there is a dose-dependent increase of free radical production. So meaning that definitely there is an oxidative stress that is stimulated by the different nanoplastic that we have tested, and that translates actually in DNA damage. So definitely the DNA damage that you can see, so genotoxicity that you can see that is dose dependent, could be linked to the fact that there is activation of free radicals that are generating within the animal. Moving on to also a very interesting endpoint, which is the microbiome and whether or not that can be affected by the presence of nanoplastic within the gastrointestinal tract of the larvae, but also in that case within the adult. And what was found, yeah, you can see now, was that there was clear differences between the different stage, so between the larvae and adult, but that is fully understandable because one is just continuously eating, whereas the other one is just having other type of activity. There was a reduction in biodiversity compared to the control in both cases, and with alteration within the microbiota. So that definitely means that presence of nanoplastic within the gastrointestinal of this model was altering the microbiome, and that may have some outcome because, as you know, microbiome is relevant for a lot of different human disease, and it's connected also to a lot of other organs. So when the microbiome is affected, you may have alteration in other organs. Moving on to the mammalian model. So in that case, first we started by one single high dose exposure, and you can see here that there was different effect at different endpoints. So all the effect seems to be type and time dependent. So we tested two polystyrene particles as benchmark and compared to our true-to-life PET, and there was definitely alteration with the colon creep depth that was elevated for all nanoplastic till 7 days, but then recovering by 28 days. So therefore, there is some recovery even after exposure. The polystyrene 50 was inducing more inflammation within or more alteration of the population of immune cells within the lymph nodes. PET was interestingly or worryingly inducing a buildup of immune infiltration within the histology of the colon and the small intestine. And overall, there was also surprising damage of the DNA, but not within the intestine cells, even though you see here that there was some damage here at 28 days at 7 days, sorry, for the polystyrene, but everything was recovered by 28 days. But the interesting bit was that the DNA damage were appearing in the liver. So therefore, not only the primary organ, the gastrointestinal tract, but also the secondary organ may react to the presence of the microplastic just for a short period of time, because here we are talking about a single exposure, and after 28 days you would expect that the material is gone, but the tissue is still responding to the first exposure. So the same as for the Drosophila, we look at microbiome. Still in the case of the single exposure with high dose. So in that case, which is not 1 week exposure, sorry, there is a mistake in the slides. So there is definitely a time-dependent alteration of the microbiome. So from the first day after the exposure has stopped, and 28 days after the exposure has stopped. And what was really interesting is that the PET that has shown also some evidence of buildup of inflammation within the colon and the small intestine was the one showing also some alteration at 28 days for the microbiome. So the microbiome was affected by the presence of the PET at 28 days compared to the control. So showing that whatever is happening at the tissue level is also impacting all the microbiome that are present within the gastrointestinal tract and that are involved in the digestion. And we also completed this single exposure showing that the PET is starting to have influence, looking at chronic exposure. So in that case, 1 week of exposure, daily for 7 days to 3 doses, all representing higher case scenario of exposure to something that is probably more relevant for human exposure. And what we found is that there is clear evidence of dose and time-dependent impact with what you can see here, that there is a column that is contracted early on. So for 7 days at day 1 and day 7, you have a shorter column size, and by day 28, the column size has fully recovered. There was also evidence of lymph nodes that were changed in terms of, so that's this one over here, where you see that at day 7, there was definitely an increase of the eosinophil presence. So that means that the animals were still reacting to the fact that the exposure has stopped. And after 28 days, so after the whole washout period, that's where you can start to see eventually long-term damage and with DNA damage within the intestine cell, even for the lowest dose. So if we conclude is that at the moment there is evidence of moderate effect, but at the same time there is a cause for concerns regarding genotoxicity or the buildup of inflammation over time. So in terms of weight of evidence, I would say that there is a strong evidence that macronanoplastics are impacting gastrointestinal toxicity. And at the same time, in terms of limitation, we will, we'll have to recognize that we have only tested one by one different nanoplastic. And obviously, what may be relevant is mixture of polymers and also associated chemicals and weathered material. And that's it. Thank you all for your attention.
Thank you very much, Cyrill.
So one of the limitations that I have is that I cannot see the whiteboard, as you know.
Okay, that's on me, and we'll try to fix it by tomorrow. But I'll share again the link on the chat. But before that, I will share your questions to the audience, because PlasticHeal speakers won't be the only ones doing the work today. So the attendants will have to work a little bit as well.
So the questions are a bit more generic. They are not only really focused entirely on gastrointestinal toxicity, and they are more in the context also taking into consideration the fact that what is happening right now in South Korea with the plastic treaty discussion going on. So I wanted to know if people are concerned by micro nanoplastic and whether or not that may change, whether there will be more hard fight for defend or reduce plastic pollution as much as possible, considering that at the end it may impact health. So there is one question about how did we stain the nanoplastic for the tea bag? So that's just by adding some dye on top of it. So it's all described in the paper. If you want to have a look, so it's just a fluorescent dye that you attach to the plastic. And that works well with, some people have tried it before as well with the Nil Red, but in the case of the work that UAB is doing, it's actually a dye that is also used in the textile industry for actually staining plastic. And in terms of combination, I believe nobody has tried within the consortium to mix different plastic within the same model. So mixture is definitely something we haven't had the time to do, even though most of us are interested to do it.
Cyrill, you also have a question on the chat regarding the size range of the nanoplastics from the PET bottle.
So I think they go from quite small, so few tens of nanometers to about 200, but the peak of so it's a full distribution. So in that case, it's not only a very narrow size distribution, and the peak is around 50 nanometer. But you can go up to 200. So you will find some fragments that are a bit bigger. But that's still within a range of what people would definitely qualify mostly as nanoplastic. Or at least within the nanomaterial community, because if we extend, the definitions are not the same.
Mm-hmm. And there's another one asking for the doses of your model systems. How do they compare with the real human exposure?
So that's a very good question, and I'm not sure anybody has the real answer, because we, as I showed you in the introduction, we don't really know what is the real exposure of humans, and that will definitely depend on where you are and what you are eating and drinking, or whether or not you are a worker in a recycling facility or in the textile industry, whether or not you go to a gym where there is poor ventilation, because that means that all the people around you that are all covered with plastic, I mean polystyrene and polyethylene kind of gym outfit, will definitely impact how much humans are typically exposed to. So we believe we are within the range of what people are exposed to, but it's definitely at the moment very difficult to know if it's true. So that's why we have, for the chronic exposure, moved from one dose, which is very high and probably unrealistic, to a very low, with a decrease of 10 over the 3 different doses to cover as much as possible the dose that we are likely getting exposed to. So sorry if I'm not answering directly the question, but I don't think anybody can answer that question. So for the impact on other organs, we haven't measured that. So probably some of you are thinking about the gut-brain axis or the gut-lung axis. So that would be really lovely to do it, but we simply didn't have the time or the resources to actually tackle that question.
Virginia, while Cyrill is asking, can you put the result of the poll in the whiteboard also?
Yes, I was waiting for more people to answer. So far,
Okay.
44 have answered. We have 44 responses. But I will just...
Okay.
So there is one question about why do you think polystyrene do not alter GIT in the in vitro model? Difficult to know, possibly because we have tried only one type of polystyrene in that particular model, but in other model where different polystyrene were play with the dose, with the charge, with whether or not there was a corona on top of it, people have identified some alteration. So it's not always the same outcome depending on the way you do it. As you know, in vitro, you control very much what you do, and therefore everything can be changing. Whereas in vivo, it's way more complicated to control everything, at least in terms of corona.
Cyrill, can I add a comment on this regarding the in vitro versus in vivo, etc.? I think that there's something to take into account here, that is dosimetry. With the in vitro systems, sometimes this is a factor that influences a lot the results. Because we have observed that even after high doses, there are certain cell types that do not internalize a lot of plastic, or that there are certain cells within the same cell culture that do not internalize the plastic. So all this heterogeneity between cell types and within the same cell line, it's a confounding factor that many times leads to wrong conclusions with the in vitro system. On the contrary, in the animal, I guess that systemic effects account for many effects, regardless of this dosimetry issue or regardless of the particle reaching inside the cell specifically.
Well, the main difference is that in most cases, at least in vivo model, we tend to look at bulk effect. So we consider all the cells, as you say, and not so much single cell analysis. Because if we were doing single cells, maybe we would be able to identify the same as what the in vitro model are showing, that some cells are hypersensitive, hyperreacting, and other cells are just not bothering at all.
Sure. Yeah, so this is well, we in PlasticHeal have tried to manage with this heterogeneity, trying to see what happens at the cells that have internalized the plastic only, and different strategies, but it's a complicated issue. So at some point, it's an important confounder.
I'm pasting the poll results on the whiteboard. I'm guessing.
Yeah, that's actually what I'm looking at.
Okay. It wouldn't be the final one because I'm guessing that more people will be answering it, but.
So what is really interesting is that people are ready to eventually stop or reduce their plastic usage, which is very encouraging because I'm afraid that's going to be difficult for all of us. But if we are not trying to do it, we are not going to reduce what's coming. Because what we are exposed right now is the accumulation of these last 80 years of mismanagement of plastic pollution. So that means that if we keep using plastic at the rate we are using it and not careful with where we put it, we are going to reach high dose. And as all of us have shown, is that the high dose are toxic. So at the moment we are in the low dose and that seems manageable. But if we keep releasing plastic and having this feedback from the environment, we are going to have an issue. Back to the chat. Check.
I think that so far you have answered the questions from the chat, right? Because maybe now we can give the floor to Steffen again so that he can present the people being part of the roundtable. But I think that we have a last question on the chat. Maybe we can take that and then give the floor to Steffen. Cyrill.
No, that's correct. With the OPEC PT, we haven't done everything as extensively as what we have done with the PET. So there is some result in some in vitro work in the Drosophila, but there will be no result in the mouse model, unfortunately. Steve?
He's jumping in right on time.
Yes, very good. So Virginia, I just need to know, do we need to have a break before we go into the actual roundtable discussion?
It's up to us. I think that people is really active right now, but I don't know, entirely up to you, Steffen.
Yeah, I think it would be good to maybe I could just introduce the panelist of the roundtable, and then we could just have a 10-minute break before we go into the actual discussions. Yep.
Okay.
Because I think we've been listening intensely to all the experts that have presented their work. So I think it'd be good to have a break before we go into the actual roundtable. So we're very pleased to have some true experts and very knowledgeable people as part of this roundtable, and basically the idea is to get their thoughts on what they think about the framework that we've presented, and also how we've used the effect data that has been generated in the project so far. So we have Birgit Sokull-Kluettgen from the JRC, who has a PhD in natural sciences, but has most importantly been involved in a lot of research and policy support work related both to nanomaterials and nano and microplastics. We have Blanca Suárez Merino, who is representing the Nanotechnology Industry Association, and has been involved in a lot of work related to both research on nanomaterials, but also policy work in regards to regulating nanomaterials in the past. We have Lars Sickert from Tetra Pak, who has a lot of experience in in the practical application of regulation from an industry perspective, and who has also been involved in several research projects on nanomaterials as a steering committee member, providing valuable input to researchers on how to make their research most relevant for experts within industry that are interested in regulation. And then we have Thierry Rabilloud, who has a combined expertise within nanotechnology and biophysics, and who is also a member of the PlasticHeal consortium, focusing on immunotoxicity of micro and nanoplastics. So I think collectively we have some very nice members of the roundtable, and we look very much forward to having their input on the framework that we have proposed. And we hope that our discussions with them can lead to a very fruitful discussion also with the almost 100 participants of this meeting today. So with that, I would suggest that we have a 10-minute break, and then we meet back here at 20 to 12. I think we should get started. So good. I'll share my screen, and then so in order to initiate the discussion about our framework, I've kind of prepared a few questions that I think are key for me as a very selfish person to get answers to and to get your input on. And of course, like everybody who's participating in this meeting is of course more than welcome to also post questions and provide comments and input and suggestions. What I would really like to do just up front is to just take around the table and have your comments on what you actually think about the framework, and if you could just maybe Birgit, maybe you could start off with just like with some of your initial thoughts on the framework and what you think of it. And then we could move on to Blanca, Lars, and Thierry to just get the discussion going.
Okay, Steffen, can you hear me?
Yes, we hear you.
Good, great. Yeah, thanks for the invitation. Indeed, very interesting. Maybe just to say for those who do not know exactly what the Joint Research Center is, it is part of the European Commission, and our work is to provide science for policy, so to advise our colleagues in Brussels who are in charge of certain policies. Within my unit, we have also the nanobiotechnology laboratory, and therefore we have experience with the investigation of particles, small particles, and that was the reason why we were also asked to look into microplastic actually. And what we did recently is we developed a methodology to measure microplastics in drinking water, which is now published in the official journal in support of the Drinking Water Directive. And we are asked to do similar work also for the Water Framework Directive and for the upcoming Urban Wastewater Treatment Directive. And so I'm very happy to follow the CASP work and also this seminar. Your tool, your framework is definitely very, very interesting and welcome, because I think it is very, very important to summarize and to aggregate in a way all the information in scientific literature we have. Steffen, you showed us how much information is there available, and also today with the different presentation, I would say overwhelmed with all the details about so many different different microplastic particles looked at, in particular polystyrene, a bit also on PET, different sizes, and so on. But if I speak with my colleagues in Brussels, for them, microplastic is microplastic, and there are some. So it is a big group of particles, plastic particles, from nano or micro to up to 5 millimeter, and in different shapes, so fibers and yeah, smaller fragments and so on. And basically, the question they would ask is it dangerous? Is it harmful for human health? And yes, can you please give us a threshold? Huh? We need a threshold for microplastic in drinking water if we want that member states really do monitoring of drinking water. We need a threshold. In the future, maybe they would ask, we need a threshold for sewage sludge and so on, so to bring it to the fields. So from this point of view, we really, really need tools which help us to summarize, to aggregate the information from science. And from this point of view, your framework is very much appreciated, though I'm still a bit struggling how really this could help in detail.
Yes,
so I stop here and leave the floor
please.
to others.
I think a very nice start. A lot of things that we can pick up on and continue to discuss. So yeah, please, Blanca, would you take over?
Thank you very much for inviting me for this roundtable. So I've been working mostly on nanomaterials, not nanoplastics. However, I think I can already see some similarities, and also in the nano world. And some of your colleagues, I think, Steffen, they have developed tools also as warning tools like the nano risk cut, and we have been involved in the Swiss precautionary matrix. So I think from those learnings, I can also see how we can have some ideas for this tool. Regarding nano, I wonder about nanoplastics as well, because in the nano field, we noticed that they are different. It's very different to have one type of nano to another. And I wonder if this is what Birgit also was talking about now in the nanoplastic field. I suppose you have different sizes of different ranges of plastics, and I think, I don't know if we can have one for all, or you could do some kind of grouping, because I don't think that you can do one threshold for everything. I guess this is a little bit like nanomaterials. Maybe you need to do grouping or ranges of groups to have threshold for those, rather than just one threshold for everything. I don't think that will work if I'm based on my knowledge of nano. So, but yeah, I agree. Policies, the policymakers, they are very pragmatic, so they need something very simple. Tell me about the risk and thresholds, I guess. But perhaps we are also scientists, and we know life is not so simple. So maybe we have to find some kind of in the middle, in between way, where it's not just one threshold, but maybe I wonder if we can have groups of threshold depending on the size range or something like this. But anyway, I will stop now, and thank you.
Thank you very much. Moving on to Lars.
Yes, thank you very much for inviting me, and thank you for a lot of interesting results. In this context, I would say I represent the packaging industry, maybe more than my company, but anyway, I think this is the legal part is, of course, very important and very interesting for us, but there is also, I think, a huge interest in this industry to learn and understand more. Consumers are starting to ask questions. Does your package have micro nanoplastics in it? Can I drink safely? How can we know? It's not easy for any packaging producer to actually know. And as we are struggling with that, what can we prove? What are the methods? How can we work further on this? And I think the framework that you presented, it gives a starting point. But my main worry is that for me and for the industry, I would say that a polymer is not a polymer. As if we talk polyethylene, I just checked, and as I found an article stating that there were more than 2,000 commercial grades of polyethylene on the market. It is high density, low density, LLD. So it's a huge, and then every commercial polymer has some additives probably. They come in different sizes. They break down differently, and they will be differently stable in different contexts. So there is a lot more to learn. And in the end, I think it's extremely complex, and it's also a world where maybe it's difficult to get the information from suppliers of the polymers. So starting with this, at least we have some starting point. But to actually reach a more easy accessible result, we probably have to go down the road of artificial intelligence, because this is extremely complex. So yeah, thank you for starting it, but I think there is a lot more to do, unfortunately.
Yes, thank you. And Thierry?
If you hear me well.
We do.
I hope so.
Yes,
excellent.
we do.
So as a researcher, I have a different perspective from Lars, of course. And I do agree that your framework is quite interesting. But for us, the main question becomes, how do we translate our results into weight of evidence? And I think there is a lot of thoughts to have at that stage about the future of this field, the development of the new approach methodologies, how we translate all of that into a decent way of evidence, and what filters, if I shall say it, shall we apply to do this? Because this exercise has been done by two other speakers this morning, and you see that the outcome they came with is quite different from same in vitro approaches. So I think we need to have a very strong reflection about that.
Okay. Yes. So thank you very much for both introducing yourself and your perspective or initial thoughts on the usability of the framework. I was wondering, you mentioned it a little bit, but are there any obvious pros and cons that you see to the framework that you think is either something that is really, really positive, or something that's kind of like really, really knee-jerking bad about the framework? I see, Bianca, you already put your hand up.
Yes, for me, one was kind of mentioned in my question, how you handle uncertainty when maybe you don't have all those endpoints, because many of those to me look like in vivo, and we are unlikely to have this information. So eventually one way may go to in vitro, but maybe sometimes you don't have all those endpoints ready, and you have to leave some gaps. How would this tool deal with gaps and uncertainty? And my other point was that maybe we are not likely to have this information from the literature. So maybe maybe I didn't understand it properly. I saw all these endpoints which look in vivo. So I wonder if you could move one level down and have more in vitro, because I don't think maybe I'm wrong, but I don't think you have all the information, which will bring to gaps, and then you have to deal with the uncertainty of leaving some fields empty. And then if you have empty fields, how does the tool? I'm not sure if that was clear to me, or if you need to still deal with that, because that will be an issue, I think. This is the first thought that came to my head when I went through your presentation. Thank you.
Yes. Thank you. So I think that raised or inspired Birgit to also say something. So please, Birgit.
Hello. So I think the framework is in particular interesting if you think about manufactured substances. So in a kind of also in the innovation of new materials, or also if you want to improve materials you have on the market. So in a safe and sustainable by design approach. There I would see really huge advantages. Where I personally have still difficulties to understand is really how we can apply it to the micro and nanoplastics pollution. So where we really deal with very different exposure in very different areas. What I was missing a bit maybe is the bioaccumulation and the persistence as a property in a way. And as for some substances, we may also not have all this information, which is on the radar plot in the moment. I was also wondering whether you could not also include or alternative include the characteristics from particles which we know can trigger toxicity, so like size and but also maybe zeta potential or things like that, hydrophobicity.
Yes,
That's
thank
all
you
for
very
the
much.
moment.
Yes, Lars, please.
Yes, and I think I conclude a little bit like you did, Birgit. There is a huge value of this categorization. And if we can have that, I think we can learn a lot from that. What are the important parameters to look at? But so if we can get the full list, I think you have a good start again, but there are other aspects of it to be mentioned. And I think that just having that is a good value. So yeah, that was my comment basically.
Would that be a full list of particle properties to look at, or is it the full list of endpoints in the radar plot that you're referring to?
Yeah, the endpoints in the radar plots, exactly.
Yeah. Yeah. Thierry?
I'm completely in line with the two previous speakers in the sense that I think that persistence is a key issue. In plastics, we are not working from nowhere. We are basically speaking of materials toxicity, and we have plenty of examples of persistent materials toxicity in the past, unfortunately. And I think we can elaborate on that. And I think, as Birgit says, that assessing persistence is key. And that may be included in the framework. And I'm very comfortable in saying that because with all the tools that have been described this morning, we know we can do that. In vitro, at least, with label particles, we can really do that, assess persistence. And I think that should be part of the studies we have to bring to the table for regulations, because that will help in making decisions. If we know material is persistent, we know we go in a certain framework of thought. If we know it's not persistent, we know we go in another framework of thought. And that really, these kind of bifurcation points are very important in trying to build rules.
But does it make a difference that we know that plastic is very persistent?
It does, in the sense that, for example, if we go into persistence, we know we have long-term effects, long-term inflammation, sustained inflammation. If it's not, if it degrades, we know it will die down at a point. And that makes a real difference. Are we speaking about having an instant poisoning, I would say, and then if we survive for the next 48 hours, it's over? We ingest a toxicant, for example, you ingest an endotoxin from bacterium, you get a good gastro, and then it's all over. Or are we going into an asbestos-like model? Okay, I take an extreme example, but are we going into that model? And the way of handling that on a risk issue is completely different.
Yeah, what I meant is that I don't think plastic would ever fall as being non-degradable. That's what I meant.
That's not true. Because that's not true. Because more and more we are trying, and here I loop back on Birgit's comment about making a safe by design approach. If we go into plastic that we hope to be degradable, is it really a solution? Do they really degrade? At which speed? What do they release? So, okay, it may be a progress. And here again, I think it's different looking into the future, going to new products, and handling the present, where I completely agree with you. At present, we are dealing with persistent pollution. Persistent toxicants, I would say. If you call them toxicants.
Yeah. Birgit, I was wondering, is there any way that you can see that we can tweak the framework so that it would also be able to deal with existing plastic pollution? Or is it something about saying that the different plastic types, we know that there's different environmental contaminants, that they're included in the radar plot, or is it a completely different starting point that one should have?
I must admit, I don't know yet in a way. It's difficult to judge. You can, of course, compare the different polymers, maybe also including additives. And if this would turn out that a certain group of polymers, or including certain additives, is of particular concern, you could do something against this. And that could be done via chemicals legislation, I think. It's and if I think about the thresholds, yeah, okay, what our colleagues have in mind is usually one threshold for microplastics. But okay, if this is not possible, one could also think about thresholds for different polymers if we know better the exposure. And also here, I think we have still big knowledge gaps, really. What is the exposure for microplastics? And what we heard today also for food and water, it's most probably very variable. So yeah, I'm sure it is a useful tool.
Yeah, no, it doesn't, like, we don't have to force feed it. Like, it's just mostly interested in understanding that, let's say we have a tool that does not enable you to establish a safe threshold or a threshold. Is it then less regulatorily relevant? Because of course, for substances where you cannot establish a safe threshold, like at least the chemical regulation should enable you to use qualitative methods instead. But I don't know whether that's accepted in your opinion, or
I don't know, I must say.
Yeah. So if we can, we should really try to see if we can facilitate the establishment of a threshold. Is that what you're saying?
Yes, that is certainly a challenge I see for the scientific community, how we establish a safe threshold in different media, food, in drinking waters. Maybe drinking water is the most urgent because we have there already some legislation. On the other hand, I'm also personally not 100% sure whether drinking water, although we are here most advanced in legislation, is the most important. So it's really, although we have so much literature, I must say there are still so many unknowns. Yeah.
Yeah. Yes, please, Bianca.
I was wondering, following the discussion, if from all these experiments that you have all done in this project, we could play around the different in vitro, in vivo extrapolation models and then get some DNLs and maybe from there build up some kind of, I don't know, some, not a threshold, but maybe I think we can have some research down that way, because I'm sure that there is a lot of information already out there, and some people are developing modeling tools for in vitro, in vivo extrapolation. Yeah, I wonder if that could be an avenue to explore as well, going down the path of thresholds. Yeah.
Yes, Thierry?
Yeah, I think with the framework you have developed, Steffen, we can reach some kind of threshold if we can expose to mixtures of plastics and additives which anticipate to be the most problematic. But then it will turn into an exposome study. Because in the sense, once you have determined the threshold, to what are the human beings really exposed? Do they cross the threshold or never? And I think that's beyond the scope of this project, of course. But I think that will be the core issue, because we see that in many additives, in many chemicals, where the producers say, oh, with my product, it's safe because you never cross the threshold, but you use this product and that one and this other one and that other one, and finally you cross the barrier. And that makes a big difference. So we have to look back into the exposure studies. That will be absolutely critical to understand better what the real risk is.
Lars, there was something else that you mentioned that I would like to just come back to, and that was like the huge diversity of the different polymers, and Birgit, you also alluded to this. Blanca, you also mentioned grouping approaches, and I was just wondering, is it like, let's say we used AI that we could generate however many plots that we wanted. Is that a way forward, assuming that we have enough information for each individual nuance of these materials, or is grouping the only way forward, you think?
Can I start? I think what the material that we have is a fraction of what is needed, to be honest. I think it is so huge, then so many varieties. So I still think it makes sense to start, but we don't get the very complete picture.
Because it's going to be extremely challenging to, like, if you say that there's 200 or 2,000 different.
For polyethylene, yes, and then I guess that there are it is huge. It is massive, yes.
Yeah, and then you have 30,000 potential additives that you could add to these. It's kind of breathtaking, I think.
And I'm sure that there is some kind of system in it, but it's very difficult to find. And maybe I can help us, but we will not get the information needed from suppliers of these polymers. But they have similarities, but it's very difficult to group them without that information.
Mm-hmm. Yeah. Anybody else has any thoughts on how we address the diversity challenge?
I have a thought, because that has been demonstrated in other fields, that if we want to make it experimentally, we'd end up with more combination than there are atoms in the universe. And that's really too huge. So we need to do big grouping in order to do that, and take some kind of characteristic or epitome results and generalize. Of course, that will be overgeneralization, but we have no choice.
I see that there's a question in the chat, maybe a little bit beyond the threshold values, but whether, Birgit, whether you could clarify a little bit about what is missing and what would be needed in order to provide useful input into regulatory processes. And maybe if you're comfortable with starting out, then maybe we could also hear from Blanca and Lars what you think, representing the package industry and the, I guess, the smaller, medium-sized enterprises. that my assumption is that you're representing, Blanca, in the Nano Industry Association. Would that make sense? To just have a small discussion about this? Birgit, is there anything about it beyond the threshold that you think is really needed from a regulatory point of view? Oh, I think we can't hear you. Yeah.
Sorry.
No problem.
Yeah. As a very concrete example, we have published a methodology to measure microplastic in drinking water, and the intention was to get more information about microplastic in drinking water in the EU, because the literature values which are available are quite diverse. And so in the moment, member states can monitor voluntary, but to force member states to monitor, it is needed to put microplastic on the watch list under the Drinking Water Directive. But to put it on the watch list, the Commission, or there must be proof that microplastic, as it is defined in the Drinking Water Directive, is hazardous and for human health. And there must be a threshold, a safe threshold. And I think this really puts us under difficulties. And it would be just great if member states would monitor voluntary, so that we get a better overview what is there, and also with time can work more towards a threshold. But maybe this is a bit too specific and goes beyond this general discussion about and what is also what I should
I think
not.
It's very good for us to understand as scientists that what your needs are. And I don't think we'll one-to-one be able to meet your needs, but at least we can look out for opportunities to meeting those needs. I think that's very good, I think. Yes, Blanca?
Yeah, no, on something else, when we have tried to seek for regulatory approval of some of other tools in the nanofield, one issue we faced was the quality of the data that was used. So I don't know if you have any way of assessing what kind of data goes into the tool. But some from the regulatory perspective, they were not convinced because of the outcomes, because of the quality of data. I think this is a different discussion altogether, but since you were talking about things which may be missing, you should watch out as well to assure that the data is good quality. And I know you have been looking at this with Klimisch scores and everything else. Now that should be visible, and because I think that gives more regulatory acceptance. That was something else that we found in other tools. So, yeah. So
if I could just ask more into that, was it basically that they said that let's say the Klimisch score of whatever study that went into your tool was insufficient, or that
no? I think it's good to have the Klimisch scores, but some tools. If I understand your tool properly, people will have to introduce the data in the tool, and then you get the graph, right?
Mm-hmm.
Is that
yes,
right?
In theory,
so,
yes.
In order to do that, because I haven't used the tool, I don't know if whoever is doing this will have to click some checkpoints to assure the outcome is good quality or not. I don't know if that's already embedded in your tool.
Mm-hmm.
You know,
yes, in theory, like the tool is an idea, but the idea would be to build a family of tools where once you upload your study, it will automatically do a check for the quality of the study. Okay. So in theory, that should be part of the scheme.
All right, so that's already because it was not
Yeah.
clear to me if because that was an issue before with other tools, so I just wanted to bring this up.
Yeah,
Okay.
but I guess you
Thank you.
like if we have 1,000 different combinations, and then you just have to work with the data that's out there, and it might not be ideal, but it's what you have. I see, Lars, you also have a hand raised.
Now I come back to that, to the industry response to consumers' worries, and our customers worry about our packages, of course. And it's a discussion that lacks a lot of information. And I think in this context that we're starting to build up a lot of measurements, yes, but it must be translated into something that is actionable. So thresholds could help definitely for us to take decisions of phasing out products that are not so good and replace them with something else. So it could initiate activities, not only implement as sorted by legislation, it's just because there is a demand for safer products.
But what happens if we can't establish a safe threshold? Is that then we just continue, or is there?
There must be something actionable, as I see it. So there must be some reasons, because we are. So how can we prove that our package is better or worse than any other package? And we can always strive to reduce micro nanoplastic to a minimum. That is of course possible, but that also could mean that we introduce something else. That could be a risk. So we need to have some more details, some more knowledge that we can use for this purpose.
Good. Thierry, see the...
Yeah, I would like to address one point on the question is what is needed to provide useful input? We need to have some kind of how do we translate the result that we have, which are mostly in vitro, into something that is relevant for regulations. And to do that, you know that in the EU, people are working about the 3R principles about in vivo experiments. And I would like to present you the CARD road to use in vitro experiment, CARD for cautious at analysis, refine and deepen. And that has been addressed in a sense by Alba, for example. Deepen is to try to extract as much information as you can by using omics approaches, for example. But they go with very cautious in analysis, because I know by experience that you can say plenty of nonsense from omic data. And refine is exactly what we touched before. Can we refine our models to do long-term exposures, assess persistence, etc., etc.? And that, if we can bring this into our mindset, then we can bring really useful input into something that will be useful for regulators. But I think that we need some kind of a process to process our in vitro data to make them as relevant as possible and not to overinterpret them.
Thank you. I see that we have a hand raised from Sabina, who is in the audience. Sabina, would you mind like turning your mic on and then pose your comment or questions?
I will turn the mic on. Just a sec. Sorry, what was the name, Stefan?
Sabina Halapadivar.
Halapadivar.
Yeah, exactly.
I don't see her.
You don't see HS?
It begins by HA, Virginia. First is the family name.
No.
I see her in the attendees.
Yeah, me too. I can see her.
She has a hand raised.
Exactly.
Hello.
Oh,
Can you
we
hear me?
you, I think. Yes, we hear you. Welcome.
Oh, okay. Thank you. Thanks.
Yep.
I'm
Would
from
you mind yes.
I'm from
Pers-
Health Canada. So thanks for this, all the presentations, wonderful presentations this morning, and they were all very interesting. And congratulations on all the amount of work that you have achieved as part of this project. I have several questions, and some of them may have been raised by others as well. But in the context of regulation, my first question is that considering that the exposure is mixture exposure, and the preliminary analysis that we have all conducted are basically on the individual plastics, how do we go about regulating single particle, single material, knowing that the exposure as well as the end toxicity is because of the mixtures? Second thing is coming from the nanotoxicology world, and I think Blanca has several times expressed the challenges that we have faced in that area of toxicology. I think we have techniques, we have knowledge and experience of how to deal with the difficult materials such as plastics, but I think it's not even good enough because plastics are presenting a new level of challenge in terms of their mixture-like composition, as well as the additional layers of chemical acting as vectors for the chemicals and other things which we have not yet started addressing actually. So considering all that, I think, do you think that we are there to already regulate these materials? My concern is that in absence of standardized, harmonized methodologies and standardized reference materials for which we have a collection of data set, and without understanding the exposure itself in humans, although we have been able to detect traces of plastics in tissues, of course, and in many different scenarios in humans, is the collective exposure good enough to cause toxicity? How do we go about even thinking of regulation? And if we were to regulate, what are we going to regulate? Is it the environmental pollution, or is it the production itself of the plastics? I'm sorry, I have too many questions, but I'm hoping that
I just wanted to thank you for your thoughts and input. And I was wondering, should we take a round? Maybe Blanca, maybe you could start off by answering or commenting on a few of the questions that you think are most relevant for you, and then Thierry could continue.
No, because one of my first questions after all these presentations was a very simple one. If you can get stable dispersions, because this is a challenge we had with nano for years. And if you don't have a stable dispersion, all the experiments that we have seen really cannot be taken into account properly, because this is the very first point we have to focus on. So, following on Sabina's question as well. And after being at the OECD, also Sabina was there in June, and there is also a new project may start in the adaptation of testing guidelines to nanoplastics, because that tells me that the guidance are not there. How can we then take those publications that you have been reviewing and accept the information that they provide or the results? This was one question I had for all the speakers this morning. I'm coming from the nano world, so I'm not following so much nanoplastics, but I also see Julia is here as well on the group. And So how can you take the data if we are still at the stage of not even harmonizing the guidelines yet? So that was a question actually for you, following Sabina's question as well, because I'm not so familiar with this area of nanoplastics. Where are we then regarding testing guidelines? So we can test in the lab. Thank you.
Okay, then I will try to answer both Blanca and Sabina's questions. Indeed, we are in a not so bad situation about dispersions, because plastics are so hydrophobic, they love to adsorb material. And in biological experiments, we have a very nice amphiphilic material which is called proteins and protein corona. And that does stabilize the plastic dispersion extremely well. And that I see a comment in the chat that PlasticHeal is using the nanogenotox protocol, and that's exactly what it is doing. So I'm not very worried about the stability of dispersion. And we have worked for now, all of us, for now 3 years with the dispersions, and that has not proved the problem. I've had much more problem with the other nanomaterials, which are even more hydrophobic and that are a nightmare to disperse into biological media. And the second, to answer Sabina's question, I don't see how you can regulate a mixture, because this mixture is not made by the producers themselves. You can regulate a mixture at the production level, saying, no, you cannot release this type of mixture in the environment. If the mixture happens after the use, and that's exactly what it is happening right now, how do you regulate the mixture? You can't. So you just can regulate, saying, for example, okay, PVC has too many additives, they are very toxic, so we have a strong threshold of PVC. That's just an example. I have nothing against PVC at all. But that's what we can really achieve, but nothing more. We can't regulate an environmental mixture by definition. So we have no other option than regulate some components.
I was wondering to just follow up on the standard issue. I think what we saw with the drinking water directive was that the Commission decided to regulate or ask for a threshold or measurements in drinking water, and without methods actually being available and without methods being standardized. And that basically meant that all EU projects all of a sudden, in quotation mark, had to include that component in the research. And it also meant that the GRC all of a sudden had a lot of activities related to measuring microplastics in drinking water, because that was an official requirement as of January 2025, or 24, sorry. So I think in that way you could use regulation to push for development of standards, because my fear, if we wait for standardization and we rely on OECD, and all the respect for the work that they do, but it just tends to take like 10, 15, 20 years before we have standards. And I don't think we really have time in many areas to wait that long before we start limiting exposure. So I was just wondering if we could also use regulation in that way. I see that Sabina, you have your hand raised. Do you want to elaborate on your questions? Are you happy with the replies you got?
Yeah, absolutely. Thanks, Thierry. And I think I agree that based on we cannot regulate mixtures and we have to go by the individuals. So are we going to then adapt a kind of approach which we have adopted for polycyclic aromatic hydrocarbons, for example? You have priority polycyclic aromatic hydrocarbons, considering that there are many plastic types which are in this mixture, and all of them are somewhat toxic. So should we go in that direction, finding a prioritized list and coming up with a toxicity data for them, and then using, maybe, I do not know if we can go as far as finding a surrogate to rank or scale or the toxicity of other materials, or if that is even necessary. But something like that we need to have, and for all of that, I would say, I guess we need to have reference materials where we have really a collection of data set from many different groups of people who are working, and then we can take it to the next stage. And I also agree, Steffen, that it is going to be 15, 20 years again, even though we have done that much of work on nanomaterials and we have so many techniques and technologies available which can be readily applied to plastics. But again, plastics is posing a different type of challenge. And we already know that some of the techniques and even the instruments are not sensitive enough to detect these plastics in different matrices. And so again, we are back to the square one. However, I think we are 10 steps ahead than where we were 20 years ago for nanomaterials, I would say. So I think there is a lot of wealth of information that we can apply, and therefore it may not be 15, 20 years. But at the same time, I do agree that we have to have a different type of approach where we can fasten the process so that we can reach the regulation more faster than otherwise.
Yes. So you mentioned reference materials, and Alba also mentioned in the chat that we needed reference materials. And Birgit, I'm going to put you on the spot, because when we discussed this on Friday, this roundtable, you also mentioned reference materials. And I think we all acknowledge that the JRC played a vital role in developing reference materials for nanomaterials. And maybe you could, is that where we should start, Birgit? The JRC will volunteer
we can start.
to...
So first at all, I think we must be careful with the wording. So reference material, for my colleagues in the metrology part of the JRC, is really giving reference for something. So for nanomaterials, for example, they had nanomaterials with a reference value for a certain size, a certified size. While what my unit was doing was what we called then, in line with our colleagues, representative materials for testing. These were the materials which were used in the OECD testing program for nanomaterials to further develop the test guidelines for the test methods for nanomaterials, because people realized that for the particles you need different methods than for soluble chemicals, for which you can use the standard methods. So for this, the JRC had this repository of manufactured nanomaterials, but here we were lucky in a way that we could just get from industry representative manufactured nanomaterials, which we subsampled and so on and so forth and distributed. But here with the plastics, we are in a totally different situation. So either because the majority of the issue, the problem, is not with manufactured plastic, microplastics. So, and anyway, this is under regulation of the REACH legislation. So this will, if not yet, it will be phased out. So what we are speaking about is really the unintentional produced microplastic, which comes from bigger plastic particles over the lifetime. And that is also why what we are exposed to, what is in our environment, what is in our food, is so diverse. And here we have really problems to make representative material. What is representative? We do not even know exactly. But even if we would go for a certain material, it is not so easy and straightforward to produce these materials. Okay, there are people who are producing PET material from plastic bottles and so on, but it's not so easy to produce it in bigger volumes. We would need bigger volumes if it would be really useful in a kind of repository to be used by many different groups over a longer period of time. Secondly, I'm also not so sure about the stability of this. So with the nanomaterials, the manufactured nanomaterials, we often had metals or metal oxides, and the stability was not such a problem. Then it could be for the plastic material, which could also be polluted by microorganisms, so that you have issues there. So it's not so straightforward and easy to make representative materials for testing in a huger amount and provide them over a longer period of time. It's really not so easy, but I agree it would be great if we would have them. On the other hand, I still believe that we need much better exposure information, and for this, standardized methods are needed so that we can really also trust that what people are measuring is more or less the same. So if different labs would measure, we would get similar results. And last not least, what also the last speaker, Cyrill, was saying, we have to change in the end the way we have to, the use of our plastic. So we have really to phase out more and more single-use plastics, and we need all over the world most probably a better waste treatment stream, so that we can ensure that less plastic is coming into the environment, and otherwise we will not be able to handle this. And today is the first day in Busan, so where big politicians will discuss these things.
Yeah, it's going to be very interesting to hear and see what happens. Sabine, I think that you have your hand raised, but I think it's an old hand, if I'm not mistaken.
One, if you don't mind me commenting.
Okay, then I'll just ask Thierry to
I would like to be a bit more optimistic about the problems that Birgit just raised about reference materials, and make some kind of Ave Maria wish that if we have many different materials, but if we provide enough documentation on each and every of them, maybe AI will be able to do this grouping and sorting out the key parameters for us. We are not good at this game, the computers are. But the key then will be the quality of the documentation, and we loop back to the Klimisch scores and things like that. Because the AI will be as good as the data are, basically. And I think it's extremely difficult, because in the plastic field, you can deal with so many different parameters. And there are always, we think of some of them right now, and maybe we miss many that will prove in the long run the key ones. But that's how our science goes.
Okay. Sabine, I didn't want to discourage you, but yes.
No, I'm just going to do a small comment. So I think most of my comments were already mentioned by Birgit. And so if we are going to I think for me, the question is plastic toxic? We understand the chemical additives may be toxic, but is plastic itself is toxic? So for that, I guess the first level of reference materials or the standardized materials that we need for toxicity analysis are the pure plastics. We need to understand if the polymers are doing something. If that is not the case, then we do have other ways of regulating those chemicals already. So we know those additives are like so many other ones that we know that they are bound to these plastics or part of the plastics are toxic. So we already have regulation system for that. So I think understanding if the plastic as a polymer itself is toxic and what type of toxicity it induces is more important, I think, in the first stage. So if we can have those pure plastics, that would be really great. And I think, as Birgit mentioned, that moving on, the question would be, they are also going to be weathered, because it's about the environmentally polluted plastics that we are talking about, which are going to be inhaled and therefore induce toxicity. So in our own case, we have been studying microplastics for the last 6 to 7 years, and we have now looking at the weathered versus non-weathered plastics, and we can see in some instances environmental weathering makes them more toxic compared to the unweathered plastics. So that complexity will come in the next stage, but I think initially we would like to have reference materials that reflect the complex universe of plastic polymers in their purity.
Thank you. Blanca, I'm gonna ask you to go before Thierry, and then Thierry, you can.
No, it's fine. I was going to start another point for the framework. So if Thierry has something ongoing for the same question.
Sabina, I'm in the same line, sorry. The point is, in that case, we should take great care about the shape. Because, for example, in the cell system that I use, the cells are extremely sensitive to shape, edges, aspect ratios, things like that. If we think we compare polymers, but at the end we compare shapes because we don't know how to produce the same shape for different polymers, the comparison we make is flawed by essence. And I would like to give you an example on what is existing in plastic right now. The process used for making the true-to-life PET particles does not make spheres. But when we buy PS particles, we buy spheres. So when we see a difference at the end, is it a difference due to the shape? Because these PET particles have edges, or is it a difference due to the polymer itself? And right now, we can't be really sure. So I think that brings another, and I know Blanca will agree with that, because it's something extrapolated from the nano world, that shape makes a difference. And it's true in the plastic too. So if we need materials, reference materials to compare polymers, we need them to be of the same shape and ideally of the same size. So that's another level of challenge, but that we must address and take into account.
Yes, Blanca, I think we're ready to move on. Yes.
Yeah, no, I was also wondering regarding the framework. I fully agree with everything that was said before. And in the nano field, we have all this issue with shapes and charge and a lot of things, which are not only the mass-driven toxicity that we are used to. But to move on from the framework, another thought that came to us was are you planning to give more weight to one endpoint to another, or to aggregate results? So whoever makes at the end. Whoever looks at the graph will have also some kind of output. Like based on this is like maybe reprotox has a higher rank than, I don't know, something else, or maybe genotox is the worst possible, or to aggregate your results somehow to provide an output. I'm not sure if you are also thinking about evolving your framework into those lines, or just to give this spider graph, which is already visual and quite nice as well. But yeah, I thought that could give some extra points to whoever is using this framework. Yeah.
Yes.
Thanks.
No, I think it's like to our initial thought was to kind of leave it with the diagram, with the plot, because we do think that we as humans are able to understand that visual expression, and that we don't need another layer of interpretation analysis to tweak it into a number necessarily. Because I think when you start adding weights and summarizing, then all of a sudden there's another layer of lack of transparency in who decides on the weights, and do we agree on them, and so on. So I think this is an attempt to keep it a little bit more honest. But then we, of course, we run into the problem that then how do we compare it with a radar plot on exposure, for instance, as you mentioned earlier, and then how do we take it from there? But that's something that we will have to think about, see if we can come up with a good idea.
Yeah. I was thinking in those lines. So, how can you compare? I know giving weights is also probably not a good idea, because why would something be more important than another, endpoint wise? On the other hand, how do you compare? Just to make it more useful. Yeah. But I know it's only a point that I don't even know how to address myself.
Yeah. I was wondering, maybe we could just take a final round and ask, I would like to hear if you have any further suggestions on how we could improve the framework, or something that we should really work on. So we already discussed the thresholds, the use of science, the standardization issues, but is there anything before we end this roundtable that you would like to mention?
I think Penny has a very nice point on the chat, because they have been using the ToxPy scores, and she knows all about this, but it's a way of kind of giving some weights. But yeah, that was actually a very nice point on the chat from Penny. Yeah. I just wanted to highlight that. Yeah. And another thing that we have seen in other tools is sometimes there is, if we have already a lot of data on a particular group of nanoplastics or something, to have it already in the tool. So it could be a default value if someone is not, missing some data, or maybe they can have a look at these kind of results which are already available. I mean, for nano, maybe for example, nano silver, we have some kind of already outcomes. So that's already embedded in some of these kind of tools. So you can already feed from that kind of the database. So I don't know if that is actually useful for you, but since there is so much data out there, I don't know if you could also embed a database there, so you can also input some data in. There's something else that I have seen in other tools that you could maybe think about.
Mm-hmm. Yes, very much so, yes. For sure. Okay. Thierry? You want to?
Well, yeah, I would make a very simple comment, because if you want to feed the framework with what we have, I suggest that you attend tomorrow's session and tomorrow's roundtable, because that will be issues that we'll deal with tomorrow, and I think that would be extremely important to click together the framework and how we feed it with our data and properly.
Yes. I think that's a very good way to end this roundtable, by making a good advertisement for tomorrow's session. So with that, I'd like to thank you very much for your active participation. It's been great. I can't believe that the time is already gone. It went very, very fast. So thank you very much. And over to you, Virginia.
Thanks, everybody. Please stay with us, because Steffen, I will also, I think that it will be also useful for all of you to stay active on the camera and on the mic. And maybe you could also take some of the comments and questions that have been written on the chat. And we can also open the microphones for the participants to, I don't know, just openly ask questions that maybe haven't been addressed so far, or comment additional issues that they want to suggest or to bring up. So I'm just like freely opening the floor to everybody. I hope it's not a lot of information at the same time, but and I don't know if somebody that has written their comments on the chat also feels that they could comment. Out loud. It appears that they are shy now.
Maybe they are more hungry than shy.
That too. I always forget that we are in Spain. That we eat at after 2. But if anybody, if no one comments, maybe, I don't know, Stephen, if you want to do some final remark, or Alba, you're more than welcome to it.
I think I'm actually good. I would lead it to Alba to maybe provide a few reflections, and then maybe we could also introduce people to the program of tomorrow.
Perfect.
Yeah, okay, well, actually, I have taken notes of all the discussion. Maybe tomorrow we can start by wrapping up all the things that have been discussed, so we can incorporate also into the discussion of tomorrow's roundtable, all these things. I was just wondering, Stephen, if it would be possible as a way of merging, like including the exposure inside the framework, is to include the exposure as a weight of evidence category. Does it make sense? Like, for instance, if there is evidence that there is a real exposure in the system you are testing, this gets higher weight of evidence for your endpoint later. So without having to separate one from the other, could it be a way of maybe attempt to include this, the exposure in the weight of evidence?
Yeah, I think it's an intriguing idea. I don't have an opinion on it, like from the top of my head. So, but it's definitely something that that we could look into and discuss further.
Sure. Well, Virginia, I don't know. For me, if the audience doesn't have any more comments, I guess that we can just let them know the program for tomorrow and just finalize the meeting, today's meeting.
Perfect. Okay, so we're at the end. Let's see, tomorrow we will shift the focus on how to make the data valuable. Highlights include talks on advanced in vitro and in vivo models by Hans Baumeister, and analytical workflows by Thorsten Remstad and Irina Stella-Lopez. We hope that the insights of those sessions are also as rich as the ones from today. If you're not able to attend tomorrow, remember that we will share some materials and summaries and the video with you after the webinar, and we will start at 9, the same as today, on the same link. So we look forward to seeing you again. Until then, feel free to use the whiteboard, which will be active as well for additional comments or questions. And we will also have a look at the chat to see if we've missed some answers to your questions or comments, and the speakers also will be able to look at them after the webinar and include some additional comments on the materials that we will be sharing with you. So I hope to see you tomorrow at the same time, and thank you for participating today. See you then.
Many thanks to all the speakers and the participants of the roundtable. Really, we appreciate it a lot, and I think we'd had a really nice discussion. It will be really helpful for the project. So bye-bye. Hope to see
Thank
you
you.
tomorrow.